to the club. Hello, everyone, and welcome to the Terasem Colloquium. I'm Giulio Prisco. I will be your host today. And I'm going to start with a short introduction on the content of today's meeting, how to preserve consciousness beyond physical death. And what is consciousness, by the way? This is the theme of today's talk. Sorry, one moment, I need to admit two people in the waiting room. And one of them is Randal. And in fact, Randal will be talking right after I have finished. I guess he will talk of mind uploading, also known as the whole brain emulation, or the science of substrate independent minds. Right after Randal, we're going to have Max. I suppose that Max will talk about cryonics. And knowing Max, I can guess that he will tell us interesting things that are ongoing in the cryonics world and what we can expect from tomorrow. Ken Hayworth will talk of brain preservation, also known as cryonics for uploaders. By the way, I'm not going to describe any of these things now because the speaker will describe them much better in their talks. We'll have Martine Rothblatt after next about mind files and the life note approach to creating cyber consciousness. By the way, Martine, I chose this picture and I hope you like it, to illustrate the point that we all have so many things in our head, and that your mind file approach consists essentially of acquiring all the things that we have in our head without having to do any high resolution, hyper sophisticated brain scan or neurotech applications. One moment, let me admit one more person in the waiting room. I'm not able to see it yet, but I hope all those I have let in are in now. And hello, everyone. And Robert McIntyre will discuss how we are moving toward the brain preservation services. And the title of this presentation is What I Learned Running a Rapid Response Human Brain Bank. Okay, we are talking of preserving consciousness, about what is it exactly? What is this thing that we want to preserve? That's a very deep philosophical questions. I don't believe anyone has a final response to that, because philosophers and scientists and metaphysicians have been discussing that for a few thousands of years, and they don't show any intention of stopping this discussion. The plan was to have Susan Schneider discuss in general terms how we could define consciousness. Unfortunately, she's not going to be able to come. She had a previous and very important commitment, and the schedule of her other commitment has changed in the last couple of days. So she sent apologies. She very much wanted to be here, but she's not going to be able to. Anyway, what she wanted to talk about was a skeptic's guide to consciousness preservation. And this is what she said to me, which can be a very short abstract of what she wanted to say today. She said, if we can crack consciousness and truly find that the neural basis of consciousness can be reproduced computationally, I would feel more confident. I do not object to the possibility, just the technological time frame and the technological plausibility. And Susan has written this very good book published last year. Let me admit someone in the waiting room. Sorry for these interruptions. This 2019 book is very good. I warmly recommend it. Some relevant things that she said in the book are quoted here. She said, there is tremendous uncertainty here due to the perplexing and controversial nature of the personal identity debate. I don't think we can object to that. She says, minds are not programs per se, but program instantiations, a given run of a program. That would be interesting to discuss. She says, I have no confidence that resorting to uploading her brain would be a form of revival. Here, she was discussing that case of the recent case of a young woman with a brain cancer who was cryonically suspended recently. So this is a no confidence vote, but she also says that the personal assistants, the Siris and Alexas of the future, might be uploaded copies of deceased humans we have loved deeply. Now, I was discussing this with Susan yesterday, and she confirms my impression that this is actually a vote of confidence to the idea of mind uploading. So she thinks some kind of mind uploading will be eventually feasible, just that she doesn't think that the upload will be a continuation of the original. And of course, we have been discussing that not for years, for much more than that, for decades. I remember the discussions about this is the original or a copy already in the 90s. Okay, let me just spend a couple of minutes to illustrate what are my essential ideas on consciousness and what it is for those who have read my book, Tales of the Turing Church. I elaborate on all these points there. I think consciousness is really part of the fabric of reality. And we are what I could call the droplets of personal consciousness or consciousness condensates. I'm not going to elaborate on any of that, just dropping some ideas. An interesting question is which substrates can support personal consciousness. For example, is a flock of birds a conscious entity? Big question mark. A flock of birds shows a lot of behaviors that one could associate with an autonomous conscious beings. So that is a question mark. Recent theories of consciousness, and in particular integrated information theory, I would say that yes, the flock of birds does show a high amount of integrated information, and from that point of view, it would qualify as a conscious being, if not for the fact that the single birds in the flock display even more integrated information due to the internal interconnections in their bodies and brains. So that here, again, according to integrated information theory, we don't have one conscious being, but an aggregate of many conscious beings. Now, from my point of view, this is something which is very much open to debate. Let me move to the end. I tend to think of personal consciousness as an attractor in the mindscape. Now, what do I mean by that? Think of the black space here as the huge, inconceivably huge thing that we could call a mindscape. That is the space of all possible states of a conscious mind. This would be a shape in a space with a huge number of dimensions, an inconceivably complex thing. And I'm borrowing the term attractor from dynamical system theory, also known as chaos theory. And here the idea is that the yellow shape is me, which means that whenever consciousness is moving on this yellow shape, consciousness is having the experience of being me. And this very complex shape encodes all my memories and feeling and personality, texture, all the very many, hugely many parameters that define me. Now, this I'm not going to take too much time exploring this concept. It is in my book anyway. So at the end of the day, I think all the consciousness preservation methods that we will discuss today, and also other, will work. I think everything that we are going to say today will work with some conditions. And these conditions are that one, we should capture in sufficient detail the structure of the attractor that is personal consciousness. And that means we should capture memories, we should capture feelings, we should capture dreams, we should capture the general texture and the look and feel that is a human personality. This is one condition. And another condition is that the target substrate that is supposed to receive the original consciousness, be it a new biological body or a robotic body or a virtual body instantiated in software or a flock of birds, whatever, must be a substrate which is able to support personal consciousness. And this is it. So let's move to the talks. Let me stop screen sharing. And since Randal is here. Let me check again that Randal is here and active. Yes, he is. So I'm going to follow the initial schedule and give the floor to Randal first. Randal, the floor is yours. Surprise us. If you have something like a PowerPoint presentation or anything like that, just share your screen.
I do, and I'll share it in just a moment. First, I just wanted to say thank you for inviting me, and it looks like you've managed to pull together a great group of speakers. So that looks really promising. I sort of wish I could stick around for the entire colloquium, but I've got work things that are going to interfere for part of it. But I'll keep on checking in as it goes on. So in any case, I hope you can hear me well, by the way. Let me know.
Yes, we can. Yes, we can.
Okay, great. Okay, then let me just make this work out with the presentation here. I did something a little different this time because of the topic. So it is true that I will speak a bit on mind uploading, but I'm trying to speak on consciousness specifically. So let's see if that works.
Yes, it does.
Lecture mode. All right, here we go. Okay, so you can all see this, right?
Yes.
Okay, excellent. So yes, I'm going to try to talk about consciousness today. Now, you'll see here right away that I put subjective experience at the top instead of consciousness, and then I called it AKA consciousness. And this is because we had a lot of discussions at Carbon Copies about how to approach consciousness and how to even discuss it and what terminology to use. One of the things that we figured was, well, there are so many different ways that people use the term consciousness that it's actually heavily overloaded and difficult to understand exactly what people mean, but that the kind of consciousness that we're most concerned with when we talk about whole brain emulation for the purposes of mind uploading is the experience that we are subjectively having. So the subjective experience, hence the word subjective experience to us at Carbon Copies seems like the one that that more directly and easily explains to people what we're talking about when we say consciousness. Now, I'm going to do a quick aside because I know that usually everybody expects me to give a quick update on what's happening in the world of whole brain emulation in one sense or another. So I'll do that after for anyone who doesn't know yet, I'll just point out what Carbon Copies is. Carbon Copies is a 501c3 nonprofit organization that is dedicated to trying to promote, educate about, and advance research in the area of whole brain emulation up until the point where it's been achieved and it can be used for things such as mind uploading. And that means we have to be a very cross-disciplinary group of people. We have a team right now that is almost 100 people strong. Although, of course, part-time because we are very poor and we don't have, full-time people working for us. And we are all across the globe, although I would say that mainly we're located in the United States and Europe with a smattering of people in Asia and Australia, and unfortunately not yet anyone from Africa, although I hear that we may be getting a member from Africa just about now. So that would be really nice. Before I even dive into anything else, one of the questions that people always ask me is, what is the difference between emulation and simulation? We've been doing so much work on trying to make our terminology clear recently that I might as well discuss this quickly. To me, the big difference when I talk about emulation versus simulation, I really think about things like, for example, a Nintendo emulator on your phone, right? An emulator is something that is emulating an experience. So it's about a purpose, an experience that you're trying to create. A brain emulation is about that. It's not about any specific detail that you implement or how you do it. A simulation is a different matter. You've got your computational simulations, which are model building. And that, again, is a way of doing quantitative hypothesis testing in science. So you have various hypotheses about how something works. For example, what is the relevant response function of this synapse? Or how does a neuron learn? And you implement those things in a model. That model becomes a computational simulation. You can test that, but also in order to create an emulation, something that is fulfilling that purpose, that achieves the success criteria for that experience they're trying to create, that emulation is going to be full of simulations. There are various simulations inside of that. So that's the difference, the way that we use it, between the term emulation and simulation. Emulation is about an experience that you're trying to create, a goal, a purpose. Simulation is about these implemented hypotheses. Now, a quick aside, still on the aside here, about my day job. I work at Voxa up in Seattle, although I'm in San Francisco, so these days everything's remote, right? Voxa builds these automated electron microscopes. There are really two branches of it. There's the Mochi system that you see on the left there, which is a tiny little SEM. And there's one on the International Space Station. And all over the place, there are lots of these. The resolution isn't fantastic, but the nice thing is they can do spectrum analysis. And so you can do things like find out why there was a hole in the Russian module recently, and whether that's because of the solid fuels having leaked into the walls and things like that. Then the other side, the other branch, is the really interesting branch, to me anyway, of what Voxa does. It's called BLADE, and it's the automated transmission electron microscopes. They don't have to be transmission electron microscopes. We're actually agnostic to the microscope technology that's used. The whole point is just to automate it, to automate sample delivery, to automate how the data is acquired from multiple microscopes and how it is then processed afterward and all of that. And so Voxa was instrumental, in fact, completely essential and necessary for the Allen Institute to be able to do its recent thing, which is this, you see here this reference to the paper, a petascale automated imaging pipeline. This is about the cubic millimeter result that they have from the mouse. This is the scanning of an entire cubic millimeter of mouse brain, which took 6 months with 6 microscopes running 24/7. And then you get an X and Y resolution of 8 nanometers, and I believe that the other resolution is something like 40. And you can see this little representation here, but we'll see more about that in just a second. This is directly relevant to whole brain emulation, the way that I look at it, at least, and I'm pretty sure that Ken, for example, is going to also be talking about that particular view of how you do whole brain emulation, or where these sorts of this sort of work goes. I just want to point out to everyone that you can look at the results yourself if you want to. At this address here, micronsexplorer.org cortical cubic millimeter, you can take a look at the results that came out of this work, because Viren Jain and Sebastian Sung, they've managed to put this on NeuroGlancer. Of course, they had a whole team working with them doing this. I just can't put them all on there. And maybe if I can, I'll just briefly jump on over there. Let's see if I can make it pop up. Oh, here we go. Hopefully you're still seeing this, even though this is now, there's a lot more detail on the screen here. This is just an opening shot of NeuroGlancer looking at the cubic millimeter data set. This is a 3-dimensional representation of the portions that have been selected and are therefore colored in. You can also just pan your way through the slices, as I'm doing here, looking at all the different parts. You can select something, and let's see how fast this is. I've just selected it. Is it going to show up anytime soon? Come on. I'm not seeing it yet. We may not have time for this in here because the connection speed matters and the speed at which they're doing the processing over there. Yeah, so I can't really show it to you at the moment, but you should look this up. You should go to this site. There are some really fascinating insights here that you just don't think of at first. So for example, because you can display the entire microvasculature easily that is around a neuron, it's even the microvasculature gives you an insight into how powerful or how important a particular neuron seems to be. The really important neurons are probably the ones that have this huge glob of microvasculature around it, providing nutrients to that cell and supporting it, whereas others have way less of that microvasculature. things that I hadn't previously really thought about, that was something that could be an indicator that you could use to understand something about the neural circuit that's there. So there's a lot of data in here that still needs to be figured out. These are just a few examples from their gallery, just in case you're interested to see what sort of things you can find in there. You can look at great detail at individual neurons, or you can look at the whole forest of them. You could try to find synapses by at least looking at how, the neurons, their dendrites and axons are kind of wrapped around each other and attempting to create a connection. We have a terminal getting very close to a spine or at least the dendrite itself. But this is still just a pile of data. And as far as whole brain emulation is concerned, this actually points out what's probably the biggest challenge coming up. In my estimation, the mapping of the brain is going fairly well. If you look at the increase in terms of the resolution and scale of what you can map, say with electron microscopy over the last few years, it's gotten a lot better. And if you look at what you can do in addition to that with optical microscopy, so that you can pick up on certain proteins and things like that, we may not have seen everything that's possible yet, but there are certainly people pushing it very hard, like all of the alumni coming out of the Boyden lab, for example. And so I have a fairly strong confidence that the structural mapping of brains is advancing at a nice clip. Obtaining a good library of all of the types of cells that are there and their synapses and their ion channels and how they all respond, the dynamics of it, that's something that is going to take longer, and it's not that great just yet. Of course, there are efforts at various centers like Genelia and the Allen Institute and many others, but there's a technological gap that still needs to be filled in somewhere there, especially if we're talking about in vivo recording. But I think that the biggest challenge is going to be this area called translation, where you have what is essentially a huge pile of measurements. Either they're structural measurements or they're functional measurements, but measurements themselves are not the parameters of your model, right? That's never the case. Even if you're just trying to do measurements of membrane potentials and you're trying to figure out what the response of a certain cell looks like, that response, when you put it in your model, it's not the bunch of measurements. You're trying to figure out what are your coefficients for certain exponentials, for example. So you're always trying to map from data that you're measuring to parameters that make sense within some hypothesis, within some neural circuit model. And that step, this translation step, is a gigantic problem. This is where there are lots and lots of PhDs and postdocs worth of work still to be done on a data set such as this cubic millimeter and all the other wonderful data sets that are coming out. So that's my quick update on what's happening on the whole brain emulation front. And now I'm going to wind back to the topic that we're at. Personally, I see that there are also questions coming in on chat. I'm not sure if I should respond to things right away. I actually usually like it when things are kind of interactive, but then there's a chance that we overrun the time. So I think I'll continue first and then we'll do that.
Just don't worry, Randal, because we are very well on time. If you want to answer the question now, please do so.
Oh, okay. Well, that could be nice because then I won't forget the topic that we were on. It says here, question for Randal, brain imaging. This is by Gunter Kletechka. How about brain imaging, not invisible light spectrum, but electromagnetic spectrum map that covers frequency between 500 Hertz and 1 Hertz? This would allow recording and saving only the neurons that are actively participating in the brain activity. These are the frequencies that the neurons send their signals about various states of our bodies, including thought processes. What do you think about this approach? Recording not only the physical location of the neurons, but also the frequency spectrum that these neurons are sending their signals. So first of all, thank you for that question. And of course, the dynamics are super important. Recording the frequency spectrum of neurons is, in fact, what happens when people are doing electrical, so functional recording of cells. That is totally normal in any case. If you were recording, let's say, just the electromagnetic spectrum, and you're not I'm imagining here that you're thinking perhaps of fMRI or something like that, that would be measuring at a large scale somehow, because you're also probably trying to co-locate where things are. Those approaches suffer from real resolution problems. It's very hard to pinpoint activity that's going on at a high resolution. But I think that in your question, the core of it is correct, and you do want to understand what the neural signals look like and what frequencies are important. And there's so many important things about that. There are the oscillations that are taking place in the brain, which are part of how the brain is putting down constraints and making itself more understandable to itself by sort of imposing a clock on how activity is being transmitted from one place to another, all of that sort of thing. So there's a lot that goes on in recording activity from neurons, either at the single neuron level, at the neural population level, or at the level of entire brain regions or the brain. And there are so many different methods that are used to do that. The reason why transmission electron microscopy or serial electron microscopy, why these methods are used here is because of the high resolution and the great detail that you get. And these sorts of things are important, for instance, when you want to understand where a synapse is. So neurons are connected to one another in a certain way, but you also want to know how they're connected, because there are interactions between these connections when they're on the same dendrite and so forth. So part of it is that we don't really know exactly at which scale we have to do the so-called scale separation. So we need to be very detailed now. It's possible that in the future, there may be less detailed ways to get at the necessary information if we understand more about brains, so that we can do abstractions earlier and still capture the necessary information. But right now, yeah, we need both the structure and we need a lot of information about dynamics to be able to build models that can be representative of what's going on. Okay, having said that, if there are other questions about this part of it, feel free to ask now or after. I do still have some slides to go, so it might be that maybe it's best to just carry on at the moment and then come back to this. Okay, so back to consciousness. And this is just emphasizing again that, yes, we decided to just point people to subjective experience. So we're building a glossary, and basically our entry for consciousness is just going to point people to subjective experience and say, look there. And that's because we don't want to confuse it with everything else. Like, you also have the medical thing, right? Is the patient awake or is the patient not awake? Yeah. Okay, so let's see what I'm trying to say here. And again, this is not a type of talk that I've given very often, so I'm probably going to mess it up a bit, but that'll be fine. Dead brains are not conscious. I'm sure somebody's going to debate this, because you can debate anything, right? But typically, we don't think of dead people and dead brains as being conscious. So this seems to point to the fact that the subjective experience isn't the state of this lump of meat at a particular moment. And yes, I know once you've been dead for a teeny bit of time, things start to decay and all of that. So, you'd have to capture it right at the moment of death or something and then say, hey, well, okay, consciousness there or not there in the state. But what I'm positing is it isn't the state of the brain itself that is conscious or that has subjective experience. And therefore, if you're thinking about Altman brains, like, in a star, you've got all the atoms arranged in a certain way, and they happen to have the same arrangement as the one that is in your brain at a certain moment, it doesn't mean that you're suddenly in that star, because there's no causal process there by which the star will, from this moment to the next, go through those states the same way that you'd be going through those states, right? So the process is missing. In other words, subjective experience is a type of signal processing, is or involves, or we can argue about that word for sure. Okay, I'm going to start talking a bit about how we actually make judgments and think about things like subjective experiences. There's a great tendency when talking about consciousness and subjective experience to put a lot of emphasis on judgments based on subjective experience or cogito ergo sum, right? This whole, but I know what consciousness is because I'm experiencing it right now, right? And I'm going to try to show why maybe that's something you can't jump to, but I'm sure everybody here already understands that. So I'm preaching to the choir. Here's just an example of a loop that might be involved, right? You get some, here's a person saying, I am conscious because I am or was aware of what you just said. And then here we have some things that are involved in this claim. There was some audiovisual input that happened. There's some language processing going on. You store a sequence of concepts in your working memory. There's some intermediate term memory happening in the hippocampus through LTP. That's got started, but it's not right there because this also takes time. It's not something that's instantaneous. There's a prefrontal executive command that confirms your experience and makes you want to respond. You check your recent memory for evidence of a perceived conversation. You compare that with previous experiences. You apply learned judgment and keep that in working memory. Then you form a response sentence, using what you know about language. And you instruct your vocal cords, and then you get proprioceptive feedback. Your own actions are also going back into your working memory loop. So this subjective experience, this process, I am conscious because I am or was aware of what you just said, is taking place in something like this, a brain-wide loop of activity that is essential to your ability to have that experience. So one thing I'm going to claim is that our statements about subjective experience should be in the past tense and not in the present tense. That you can really only say that you became aware of the fact that you were aware of something, not that you are currently aware. Because the loop involves all of these buffers, all of these memory buffers, and the comparisons that you have to make. And you can only compare things if you're holding them in a buffer of some kind. So the comparisons that you need for these judgments that you had a subjective experience depend on memory buffers. Now, what do we know about these memory buffers? So you're already seeing where I'm going with this. Memory is essential to subjective experience and everything else that comes from that, like personal identity and all that. So these buffers, they need to be functionally integrative. They have to be able to use various types of input, various types of processing, various types of activity in the brain, not just visual or auditory, but many kinds. They need to be temporally integrative. So they have to define episodes so that you have a perception of something having happened. They need to capture instantaneously because synapses don't modify that quickly. So this cannot be memory that depends on synaptic modification, at least not to a great degree. And I guess what we want is there should be measurable evidence of them during cognitive performance that we think is awake and aware. So let's have a little look at these short-term buffers because those seem to be essential for that. And I'm just going to pick on one model of those that I happen to be very familiar with and that I like a lot. This is the model that's ultimately based on work that Lissman did quite a while back in the 90s, and that his students have taken on from there, and others as well. And it's the idea that you can maintain exactly the number of items that we usually have in working memory in a set of gamma rhythms nested in theta rhythms in the brain. And these rhythms are there for reasons. Like the theta rhythm is basically being imposed both by intrinsic properties of the cells as well as by input that's arriving and giving you a theta clock. And gamma rhythm is being imposed by the recovery time needed after this recurrent inhibition suppresses all activity after a set of pyramidal cells or principal cells is fired. So you've got these two rhythms that are there due to intrinsic factors. And these rhythms give you a kind of clock, and they give you a way of having separated patterns of activity happening in a consecutive order, an order that is maintained over time. So you have an episodic buffer that keeps those activities going for a while. And you can see where that's going. This can happen in all of or most of the principal cells of the cortex, for instance. A lot of those have this ADP after depolarization, which allows them to keep on spiking once they fired. And this happens not in all modes, like not in various sleep modes that you're in, but specifically during awake, what we normally consider conscious activities. And this persistent firing allows for these sorts of buffers. There's a lot of other work that goes into this. There have been many, many papers written about it. So I'm not going to get into all those details. But the point is that this is at least one model. There are other models of how you can have a limited length working buffer or short-term buffer that does not depend on specific synaptic connectivity. It does, of course, depend on there being synapses through which you can propagate activity, and that these can be used for the purposes that we're looking at. So I've already discussed that after depolarization is important for this particular model here. And here the important thing is also that this allows for phase offsets between regions, where you can have that in that rhythmic activity, that in one region, the cells are becoming activated and more ready to fire, just as in the other region, they are less active, so they won't have interfering patterns, and they're ready to acquire, to receive information from the adjacent region. So there's this phase offset method. You're seeing here how the brain is going about the business of making itself quite less chaotic than it might otherwise be as an analog system, more like a computer that has modes and is doing things, handing off information from one region to another. Now, there are some other interesting aspects, like the hippocampus that is involved with this, with memory that is established from these sorts of episodic buffered pieces of activity that then get turned into LTP, that these hippocampal cells don't seem to be specific to a particular type of memory. They are reused when you take a rat, for example, through multiple experiences over days or weeks. You'll find that the same cells are reused for entirely different patterns about entirely different things that they've experienced. So it's like there's this pool of resources that can be used to do this task of building these episodic memories, and do the temporal integration as well, so that it doesn't matter whether you're experiencing something where each item in the episode is happening at a second different distance from one another, or millisecond, or within certain limits. So there are areas of the brain that are involved in this, and that allow you to have these working memory experiences, and they can be from all over the brain. So that is there. And I often like to think of this, this is not my idea. This is something, of course, that others have also come up with, as say the other side of this pyramid that we often think of when we talk in terms of deep learning or something, where you'll say, okay, the neocortex is like this pyramid where you've got features on one end, and then the features get combined to more complex things and so forth. And you're going in that one pyramid. It's like there's this other opposite one that is all about these oscillatory buffers, an inverse pyramid conjoined with that one that maybe is still missing in parts of what we see in AI today. I'm not going to go into this. This is too much self-advocacy. This is just work from back at Boston University about those buffers, too much stuff. Another thing I really like is, of course, like everybody, the discovery of grid cells, and now things like, one of the last things that Howard Eichenbaum did was discover time cells as well. But yeah, grid cells are wonderful in a way. In rats, they're used mainly to map out space instantaneously, and to be able to then generate place cells that allow the animal to move around and find something or go somewhere. But really what they are, at least I think, and some others also think, in humans is not so much place related, but concept related. So you can take any kind of input, throw them into this grid cell generator that is, for instance, in your medial temporal lobes. And then on the output side, you can have something that is a tensor, a place somewhere in a multidimensional space mapped out by these grid cells together. So you get a way of instantly mapping entirely novel patterns and sets of patterns that you're putting into the system. It's a concept generator area. And it's interesting to me that that's likely also the place where there's still adult neurogenesis. I mean, there might be a good reason why that would be a useful resource for an area like that that's trying to generate novel concepts without interference from previously existing patterns and established connections. But I'm not sure about that, because there's still some debate about whether all of that adult neurogenesis is quite what we think it is. So back to something a little bit more abstract here. So what does a self model need? What we can see when well, this is more introspective, really. I don't think we see this from the models, but it appears like we don't really need awareness of detail in two ways. I'm not aware of the functional detail, like I cannot tell you which synapse was involved with some thought I just had, or realizing right now that I was aware of giving that last bit of the presentation here. No idea what synapse was involved. I also don't know all the details anymore. Even now, at this moment, I would not be able to repeat that previous experience to you in detail. So the details I don't have access to. But what it does seem to need is it needs to have some concept level flags that allow us to make decisions. And it needs to be able to include both the external experience, external activity, and that's, you know, cognitive activity generated by external input, and also internal things like plans, goals, drives, and these things all having been pushed through the filter of experience, pushed through our particular memory makeup that is giving us a particular type of plan, a particular type of goal. But you'll notice that neither of these things that I've just mentioned really say anything about, oh, it has to, you know, the self-model needs to give you an objective and pure and correct version of what the world is like, or what you yourself is like. I haven't seen that yet. I haven't seen that that's in there. I'm sure there's some reason to have somewhat of a correct model. You need to know that you have two arms or two legs so that you can make sure that they don't always get caught between doors. But yeah, how much detail? So illusions. I really, really like talking about illusions, and I like hearing about them because I think it's so useful to talk about illusions in order to get past preconceived notions of what something is. And of course, we've had this already in the study of vision. You know, visual illusions tell us a lot about how visual processing happens, and therefore the things that we actually do work with, the data that we have and the data that we don't have, and what the mind makes up based on something. And it's similar in other areas. I like some of the recent, you know, I guess I'd almost call it just statements or claims that are made, for instance, by the neuroscientist Anil Seth. This is paraphrasing, but he basically says that we're only aware of that which we can compute. And because that cannot include the whole of the concept that we try to perceive, there's always an illusion involved. And he gives this example here that kind of connects this to that loop I was talking about before that we're always in. He says, for instance, we're fearful because we perceive our heart is beating faster. Our survival mechanism to ready us to respond to a threat picked up by the visual cortex, for instance. Our feelings, even much of our experience of free will, are also hallucinations issued by the mind to control ourselves. So part of what we think our experience is of reality and of ourselves is a response to unconscious things that are actually happening within ourselves. And consciousness seems to be just a thin veneer that is put on top that's giving us some sort of a control loop or some kind of inspector that we can work with. So using subjective experience as evidence for objective reality, to me, feels like trying to let a player in the game, you know, in football, be the referee. It's very dangerous. And I mean, I'm sure this is something that is recognized all throughout science, that it's better to have an objective measurement and to keep on testing your models than to try to introspect. That's where that departs from metaphysics. Or from, I mean, metaphysics is also about more. Metaphysics is often about logic, for example, which isn't just a matter of, you know, introspection and trying to say, oh, what I'm experiencing is what's real. What you're experiencing is realish, as in certainly there's something there. I'm not saying that there is no such thing as consciousness or subjective experience. I'm just saying, just like with visual illusions, what this means is it's probably actually different than what it seems like, right? Now, a quick bit of advertising here. Carbon Copies is co-organizing an entire neuro track with Foresight over the next year. So we're going to have keynote speakers every month, and we're going to have 4 workshops. One of these workshops is going to be on illusions. And it's not just a magician workshop, although I'd love to have a magician there just for the fun of it. It'd be great. The real purpose is to try to go from things like visual illusions and take people through that process to realize that you need to be very careful about what you think something is, because you get all the way to the illusion of self and personal identity. And again, I'll say, by illusion, I do not mean it doesn't exist. I'm just saying it's probably different than it feels or seems. And okay, quick examples. So here we have just trying to take the computational neuroscience approach to 3 examples here. What's actually happening when someone has a subjective experience like, ouch, that hurt? Let's see if this pops up. This one here is input driven, right? Something hurt you. There was a noxious stimulus that impacted your skin. There was some peripheral cell nerve firing. The fibers went to the central nervous system. There's some subconscious countermeasures already start, and you're getting an adrenaline flood. There's blood pressure is raised, heart rate accelerated, motor cells in the extremity are activated. You're beginning to withdraw before you even realize what's going on. So you're countering the pain already. Now in yellow, I'm showing the things that rise to subjective experience, to awareness. Your neocortex is processing the sensation. It's like in one of those short-term buffers. It's associated with previous experience. They're trying to see what the hell's going on here. There's attention being modulated, and you're figuring out which part of your body's involved. There's a proprioceptive input of the response that you're doing. The fact that you're pulling away is also immediately becoming apparent, and so forth. It just kind of goes on until you have like this plan eventually where you express yourself and go, ouch, that hurts. So you can see how that again is in a loop like that. Let's see, is this the other one? Yeah, it's the proprioceptive loop one. Okay, so this one, I'm calling this one driven by the proprioceptive loop. There's also an external input involved actually. So maybe this isn't quite right to call it different. So in this case, someone is getting kissed on the cheek, and there's a blueprint sort of response that begins: hormones, adrenaline, heart acceleration, blood pressure, upregulating certain neurons, and lights seem brighter, sounds, you're more sensitive to them. You change your posture, and then you become aware of that proprioceptive input, of the sensations on the cheek and your reactions to it. And the sequence is getting into your buffer again, and you're beginning to react to it. You're comparing it with known self states, and then you realize, I'm excited. Let's see, is it coming up by itself? Click again. Okay, the last one. Let's say this one is, let's call this elicited by internal modes. You're just thinking about something, you're busy with something, you have a plan, you're driven by something. You're working on something with your working memory, and you're retaining sequences so that you can do some problem solving, right? You need to be able to compare possible solutions and things like that. This again is sort of a back and forth between conscious and unconscious things. And you have, again, judgments about what's going on while you're going through it. And even just coming up with a solution for your problem is triggering dopamine. So again, you've got this thing that is already built in that's going to excite certain neurons and give you this proprioceptive feedback, by elevating your heart rate again, by making your cheeks flush and all of that stuff that is telling you what you're experiencing at this moment. And then you get this result and you think, oh my God, I have a great idea. So you're seeing, again, the subjective experience, this model, the self model, it's contained within this whole other thing. And it's not as if it's just all by itself aware that it has a great idea. The idea is only great because you have all these other sensations going on. And you only really get there by going through this loop. So it's this part of a bigger whole where a lot of buffers, a lot of activity is involved. Something that's missing from these is there's no holistic self-experience. There's nothing you can just detach from the rest that takes the whole in count, but that is somehow separate from it. So you neither have this holistic self-experience, nor do you have a distinct thing that is the subjective experience of it all. At every point, the patterns of neural activity that are involved there, they're triggering other patterns of neural activity. It's this whole flow that's involved in the brain. They're all there as part of that subjective experience. Oh, I pressed the wrong button. Sorry, I have to go back down there. There we are. I'm almost at the end, by the way.
In fact, I suggest that you accelerate because you are almost at the end indeed. And
okay.
we have, and we also have a couple of questions for you in the chat.
Okay, then let me skip this one because this is you can look at this elsewhere. The main takeaways of this are that subjective experience is a process, not a momentary state. So that's what I'm claiming. That subjective experiences are post hoc. They should be made as past tense statements, intrinsically linked to memory. And that subjective experience is limited by the types of memory that it depends on, such as short-term memory. And that's something that I think you can discover in a scientific way. And I'm not going to ask this then, but that was my bonus question is, if you could from this consider what mechanisms there might be, why we have obviously this feeling of a homunculus, why it makes us almost by definition dualist to be living in something with a self-model like that. And then I'll just thank the people that I was talking to who really gave me a lot of insights that helped this branch of what we're thinking about grow. We haven't done a lot of events or talks about subjective experience yet, so maybe there will be more about that soon. And of course, thank you to all of the team members at Carbon Copies. Thanks.
Thank you very much, Randal. And now if you could say something about the questions in the chat without taking more than, let's say, a couple of minutes. All
right, let's see. Oh, wow, I see that you were trying to warn me about time in there. Okay. Yeah, the tensor analogy. What would represent the principal direction of the tensor? I don't have an answer for that. I'm sorry, so I'll just skip that one. Okay, Anil Seth, it's another recommendation to look at that. So I think David Wood agrees that his work is interesting. Brent Alsop says, substrate independent, you need a dictionary to know what red, pain, and pleasure mean, while the quality of your red, pain, and pleasure are just physical facts. Those physical quantities are the definitions.
Yeah, you mind if I show the image?
Yeah, Brent, would you like to? Yes, please do. Yeah, I think that would be very useful.
Okay, let's see. This guy. Share. There, can you see that? The one on the right is substrate independent. The other two, the quality of one's knowledge of red has a physical fact and the quality of the other one's has a physical fact. And those are the definition of abstract words, red and green. Does that make sense? To me, that describes what consciousness is, that single image there.
What's the claim here?
The quality of your knowledge of red has an intrinsic redness quality. That's a physical, objectively observable fact. The person in the middle is different. He represents red knowledge with a different physical quality. The one on the right represents red with an abstract word red. You need a dictionary to know what red means. You need a dictionary to know what pain means. Yeah. You need a dictionary to know what pleasure means. The quality of your pain and pleasure is a physical fact, objectively observable.
Is your red the same as mine, or is a rose the same color to everyone? You know, is the red?
And you could engineer two different beings to be. Yeah, you could engineer two different beings to be those two different things.
Yeah, I've had interesting conversations about that because there's a lot of this. People make a lot of this notion that someone's red's not going to be the same as someone else's red.
You could engineer
someone. I'd like to debate that. I would like to have a workshop or a talk or whatever in which I will claim that your red is essentially the same as my red, and basically discard most of what qualia stuff is about. And the reason is this. We never just talk about red. The concept red, we never really work with just red. We always work with red in a context, like we'll think about a red rose, or we'll think about a red traffic light, or we'll think about a red pen, or something like that, or red blood. So for us, red is always in a certain context. Now the robot on the right, if it's learned all the same context, it's got all the same memories about how the world is put together, and you're giving it the input from that visual, it's going to have essentially the same red experience as someone else who is talking about it in that context. This is how we can communicate. This is how it's, it's very much the same for us. This notion that red is green for some people, I don't buy it. I mean, if they're seeing a traffic light and it looks red, it's red to them.
No, I'm talking about those are engineered systems. You could engineer each one of those different systems to all function the same. They could all pick the strawberry. They could all function the same. You could engineer.
I will point out briefly here that blind people exist.
Oh, sure. Yeah, so some people are not going to be part of that particular conversation of what the traffic light looks like. Because
again, if I can interrupt, my experience with this specific discussion is that it really tends to go on forever with no agreement ever reached. That's why I would suggest to stop this specific discussion here. Maybe we continue by email.
Thanks, Brett.
And since we are 5 minutes past the hour, I'd like to thank Randal very much for a great talk. And I would also like to ask Brent to stop sharing the screen. Okay. And next, I'll give the floor to Max. Over to you, Max.
Oh, thank you. It's great to be here. I was just thinking while I was listening to Randal that I've been thinking about these ideas for an awfully long time, over a third of a century, I realize at this point. And it's kind of wonderful because back then this was something that really was discussed by a few philosophers and then a few neuroscientists, and now it's becoming something that's discussed much more widely. And we've got some really smart people like Randal and Ken and others who are actually doing important practical work on this. So it's pretty exciting to see this develop, because back in the early days of X-Ray magazine, people said we were crazy, to use the old expression, but now we're seeing all these ideas being discussed and actually researched. So that's pretty good, great, and thanks for organizing this event. My talk is not going to be as organized as Randal's, a bit more of a stream of consciousness, or I guess I should say a stream of subjective awareness, if that's better, Randal. I'm not actually don't talk too much about cryonics, I will come to that, but a bit of philosophical background for those who are not familiar. I see some names that probably don't know my work. I actually wrote a doctoral dissertation back in 1995. It was published on called The Diachronic Self, Identity, Continuity, and Transformation. And so I'm going to kind of just briefly go over some ideas about personal identity as it relates to continuity of consciousness in the future. Because it's kind of the philosophical background is kind of important for the practical work and for what we can do to try to preserve our consciousness in the future. And I'm not going to go to great technical detail on the philosophy, but just some broad strokes. So really, everything Randal said, I'm in agreement with. So that's a good start. I think that we're on the same ground there. There are some areas that get a little bit tricky, but basically the idea is that the self or the person is basically a pattern running on matter. It's an instantiation of a certain pattern running on a material structure. It's not something that exists independently of all matter. So I disagree, for instance, with Hans Moravec, who, at least in some versions of his work, seems to suggest that just by applying some kind of mathematical representation, interpretation, you can kind of create a personality, even though there's nothing actually happening physically. It's just kind of a mathematical interpretation of anything like a star or a planet. That seems to me to be very kind of strangely Platonistic. So I think we do actually have to look at the actual structure of matter, the process that's going on, and all the work that people like Randal are doing on that, and all the neuroscientists. So mind stuff, basically, if you want to use the term mind itself, is a little embarrassing. It's kind of outdated, but it's a term that everybody uses, or consciousness, which also has many problems, as you've just heard some of those about. We're not really that conscious, I think, and as those of us who think of ourselves as transhumanists realize that consciousness is not something we either have or we don't have. There are degrees of consciousness. That's quite reasonable, I think, to say that animals have degrees of consciousness. I think it's also quite reasonable, although perhaps unpopular, to say that humans are not very conscious. I think we're actually fairly minimal in our consciousness. Yeah, we have more consciousness than an amoeba or a slug, but I think compared to the kinds of beings that we could become, or that might come up in the future, we'll be seen as really largely unconscious, that we don't have much idea of what's going on in our minds. We don't know why we're feeling certain things. We make up stories about our motivations. And we're really pretty crude in terms of the consciousness. It's just a very surface level thing that's kind of inventing stories about what's already happened. And I agree again, as Randal was saying, that really consciousness is not a present tense thing. It's really what's happened, and you're kind of evaluating processes that have happened in the past, hopefully the very recent past. So that's one thing I think is kind of an important foundational idea, that to have mind or personal identity, you have to have some kind of physical structure that embodies that. The second point I want to make, which I think probably not be controversial here, is that dualism is almost certainly false. And I'm not going to go through all the different arguments or even try to define dualism too much, because people have often called transhumanists dualists, which is unfortunate because they clearly don't understand what dualism is as distinct from, say, functionalism in its various forms. Dualism in the classic sense really is that there are two basic different substances in the universe. There is matter and there is mind, good old Cartesian dualism. And then, of course, all the problems of how those interact. And it really is kind of a dead end. So one of the main alternatives to that in philosophy is functionalism, which really says, again, that you have to have some kind of physical structure to embody mind or personal identity, but it can take many different forms potentially. Not every possible form, perhaps, but it can take many different forms. It basically underlies the idea of substrate independence. There's different kinds of functionalism. Again, don't really need to go into that, but the basic idea is that your thoughts, your experience, your memories don't necessarily have to be instantiated in any specific physical structure. You could maybe alter the matter embodying the neurons in your particular brain, and as long as the function's the same, you would have the same experiences potentially. And that potentially could be the same for going to non-physical, non-biological structures as well. Although there things get tricky, because we have to then think about what level of abstraction we're talking about. We have to model the neurons. Do we have to model subneural structures? Can we do away with all of that in favor of something on a kind of a higher, more abstract level? And I'm not going to go into those details too much. But for anybody who likes thought experiments and philosophy, my dissertation is actually online, The Diachronic Self, and it's a lot of fun for an area of philosophy, because you can do all kinds of experiments and try to tease apart the various components of identity, what's essential and what isn't. I'm basically kind of a Parfitian in that I think that what makes you the same person over time and what it is you want to preserve about yourself is essentially psychological continuity and connectedness. In other words, it's not the particular matter that you're made of. It's really the way that your values, your goals, your beliefs, your projects, your feelings, your personality, how those traits are connected between any two periods of time, and how rich those connections are. And there has to be a certain degree of connectedness and continuity. So there's a huge disruption. For instance, if you have walk out into the street, get hit by a truck, and you have brain damage, and you lose all your memories, your personality changes drastically. In my view, even though legally you survived, I may not be the same person. I may be gone. If I completely changed personality, have basically very little connection to my previous self, I wouldn't consider that a form of survival. Whereas if I was, if my heart gave out for some reason and I'm preserved in some way and brought back in perhaps in let's just say an android body made of something different than our usual biology, if it preserves all my psychological structures, so I remember my past life, I have the same values and beliefs and so on, then in my view, I have survived. That's what matters. Now we get to some tricky issues, because for a long time in philosophy, this is called the problem of personal identity. Now, I don't really think identity is what matters. I think that's kind of a mistaken pathway that people go down, trying to argue whether identity is being preserved. And identity is really more of a logical construct, and I don't think it's what matters for survival. I try to make that at least vaguely clear. To think about the logic of identity, identity is transitive. So let's suppose we have one person, we have me today, we have me, Max 2021. Let's imagine that somehow I fission into two individuals, and let's not worry too much about the details of how that happens. But now we have two people. We have, so let's say Max A here, and then Max B and C. Well, B and C are clearly not the same individual, because we can see that they're different, separate individuals. So logically, they're not the same person. So then you have to think, well, the logic of identity is broken down. So if they're not identical to each other, maybe I can't be identical with either of those. But is that really what matters? Have I died? No, I really haven't. I've fissioned, I've become two. it's a lot better than dying. There will be certain complications. First of all, those two Maxes are going to have to fight over who gets my wife, Natasha, or I guess she makes the choice. Maybe one of them came out slightly better than the other one. They're going to have to decide who gets my property and who gets to pet the dogs and so on. So there are a lot of practical complications involved in that. But I've survived in those two new individuals on this view. So I don't really care about identity in that logical sense. I care about continuity of what makes me the person I am. So that's what allows something like substrate independence, the ability for us to potentially continue. And again, I don't think this is going to happen in any time in the near future, but potentially we could continue on a different kind of platform. I also very much like, by the way, the definition of emulation and simulation. I agree with that, and I think that emulation is really the term we should be looking for. One other outcome of this general philosophical view is that survival of personality is not all or nothing. I think this is another common mistake we see in the way people typically think about these things. They, either alive or dead, you've either survived or you haven't. And again, you could have an accident where there's a lot of damage done, loss of memory, some change to personality, and then in some sense you've kind of partially survived. It's really a matter of degree. It could be such a drastic change from the kind of change you get from overnight, where you kind of change just a little bit, but you've got very strong connectedness and continuity with the day before person. If that continuity is greatly disrupted, I think there isn't really a right answer as to whether that's still you or not. It's a matter of degree. So we can say it's partial survival, and that becomes relevant when we think about things like cryopreservation or uploading, which may or may not be perfect in the fidelity. And you can ask yourself, well, how much fidelity would I want before I would consider this to be a waste of time, that I'm not really surviving enough? So it's not whether I survive yes or no, because I don't think that's really the question. It's how much do I need to survive? And then you get into some interesting issues. In fact, in my dissertation, I kind of dig into the components of identity and continuity. And I think there's some differences between me and some other people who I share a lot of thinking with, in that, for instance, I don't personally find memory to be that important. And maybe I should say decorative memory. I think obviously the kind of memory, if you want to call it that, involves my skills and my ability to do things. I think those are important, but decorative memories actually, to me, aren't that important. I think I could lose most of those and I would still consider myself to be surviving if I had the same personality, the same drives, and so on. Other people will consider that to be pretty drastically a loss of self. Maybe the reason for that is I don't have a very good memory. I don't remember much about my early life very well, and I don't kind of review things very well to put those into long-term memory. But I don't feel like I've died over time particularly. I just don't remember stuff. I think it's more important, the kind of person I am and how that's affected who I am and what I will do in the future. But that's one of those kind of interesting questions: is there a right or wrong answer, or is it a matter of what you value in terms of survival? We might want to think, well, there must be a right answer about have I survived or not, or how much. But it's a little bit tricky. Maybe it depends on what you value. That's kind of an open question to me. So I won't go too much into those questions of how much you can change, what kind of disruptions you can go into. If you do want to read my dissertation, which I'm told is relatively entertaining for PhD dissertations, it is online. I go into that question. I have a whole chapter on when does continuity end? In other words, what really is death, which is an important question for those who are actually looking into ways of preserving continuity of consciousness. Death isn't what we think it is. There isn't just life and death. There's kind of an in-between state as well, where you've preserved the structure, but there's no processing going on. To refer back again to what Randal was saying, you have a structure there, but you're not conscious, of course, unless it's running, if it's actually processing something. So to sort of switch from the philosophy a bit more to what we can actually do to ensure survival of consciousness and identity, I think that right now there is basically one way you can do this, with maybe another method that is potentially workable. If you don't make it to the time where we fix the aging problem, and I think probably everybody here agrees that is a problem that needs to be fixed, I have to say I'm a little pessimistic now. I'm 57 years old. I've been watching life extension stuff for at least 40 years, and I'm not seeing a whole lot of progress. I'm not seeing anyone beating Louise Calment, who lived to 122. Nobody has done that. In fact, nobody's even come close within about 3 years. And that's not seeing any progress. There's some little tiny progress around the edges in terms of regeneration of the thymus, which I think is pretty interesting stuff and helps you not die early. but I'm not seeing anything that's extending the maximum lifespan or really helping us get beyond 100. And that's been decades. And yeah, of course, you've got the strong singularity contingent to say, oh yeah, but you're just not thinking, you're just thinking linearly and not exponentially. Well, I'm getting a little tired of that explanation because we've been on this low part of the exponential for a long time now. So I think realistically, yes, we don't know what the future holds and what kind of breakthrough might happen. It could happen suddenly, very unexpectedly in the next 3 or 5 years. But I'm thinking that there's a good chance at my age that, maybe I have another 40 years left. There's a chance that that problem will be solved, but there's a very good chance that it won't. So I'm kind of reconciling myself to having to be preserved in some way to be revived later. And I would urge people who are, of any kind of age to take that very seriously, because you want to put some steps in place. Plus, of course, bad things happen, as I can tell you from being involved in cryonics. Plenty of people don't make it remotely close to 97. So it's always good to have a backup plan. So what is the backup plan? Well, I think the answer is basically the best backup plan we have right now is basically cryopreservation. There is, of course, also the possibility of chemical preservation, and then there are some hybrid options too. But those are not currently offered by organizations that actually make that happen. That may become true in the future. So your basic option right now is cryopreservation. So for those who are not knowing everybody on this call, let me just briefly talk about that a little bit, because this is something you can actually do today. Right now you can't upload yourself, right? We can't do a good enough scan and upload you. But if you're cryopreserved, if we've done a good enough job in preserving you, if uploading is what you want, then you have the material there to be scanned and uploaded in the future. That's not our basic scenario, by the way, because there are philosophical issues. Again, some people will not see an upload as their survival. They'll see it as basically a copy of them psychologically. Now, I don't actually agree with that. I'm quite happy with the upload scenario myself, but I know that many people sign up for cryonics are not comfortable with that. They think, well, that they're gone and they've been replaced by someone who's very much like them on a different platform. So that's not a base scenario, but I think it is a possible one. But unless you're there, unless you have a brain intact to scan in the future, of course, there's nothing to work with. Unless you can do some amazing reconstruction from historical information, which would be a bit of a stretch in my view. So I would suggest that something like cryopreservation arrangement should be part of everybody's portfolio of life extension options. And I use the term portfolio here based on how people look at investing. If you're looking to invest for your future and to have continuity of your wealth, you want to have investments in a number of different areas. Some things that are more high growth or maybe high risk, things that are safer, but don't yield as many returns. And you kind of spread it out over different options. Well, so yeah, you want to do thymic regeneration. You want to take any supplements that maybe might work. You want to do anything that might make you healthier, extend your life. But as part of your portfolio, I would say cryopreservation is pretty important. Well, do we have any reason to think that cryopreservation actually will preserve your structure well enough to give you a chance of being either revived in your physical body, which of course would be your de-aged, very healthy body, or uploaded, or revived and then uploaded? Well, I think yes. I think there's actually pretty good evidence already out there, and there's even better evidence that should come out in the next year. First of all, at Alcor for the last 10 years or so, we've been doing CT scans of our patients. And once we have our CT scanner back in place in the organization, we will be doing scans not only of neuro patients, but whole body patients, both before, during, and after cryoprotective perfusion. And what we've seen from the CT scans so far, we've done a few dozen of them, is that there is a wide variety of outcomes. In some cases, it doesn't look good. People who we don't get a warning of impending legal death for quite a few hours, it doesn't look so good, or if people have an aneurysm or some other problem like that. But in other cases, we have some really good outcomes. For instance, Fred Chamberlain, our co-founder, he moved from Florida to Arizona, right near the facility. We were on standby right by his bedside. We could begin within seconds of legal death being declared. And since Fred had a good vascular system, he got a really good cryoprotection. And the CT scan showed basically no ice formation visible at all. So that's a pretty good outcome. Now, CT scans are not sufficient to tell us what's going on the level of detail that we need to know if memory is being preserved. But we have some other information too, apart from, Robert McIntyre, who'll be talking later, can talk about the aldehyde stabilized cryopreservation and the results of that, which look stunningly good. The issue with that is whether we can reverse that. So we're kind of looking at actually achieving images that are just as good in electron microscope studies as that, but without doing the cross-linking. And I think that's something that's going to be very important in the research in the next year or two. That's one of our major projects. But what we have seen is we do have some electron microscope samples. And one of the issues has been, and that was really the point of Robert's work, I think, or part of it, is that when we do cryoprotection currently, the brain tends to dehydrate and it shrinks down, and that makes it quite hard for light to pass through. And so you get kind of pretty dark images that are hard to interpret as to how well you've done. And what Robert did basically was to stop that brain shrinkage using stabilization. But again, one issue with that is, how do we reverse that? So by using a certain new treatment that's in the lab right now, we might be able to actually prevent that dehydration and produce as good images. But we do have some images, and I can't say too much about this because it's not a published paper yet, but a certain very famous cryobiologist, who many people will know the name of, is being pushed pretty hard right now to finish something he's been working on for 4 years, which is to publish a paper on a case we did a few years ago, in which we took brain samples, essentially, and did differential scanning calorimetry, EM studies, and a number of other different kinds of analyses. And from what I hear, the results are pretty damn fantastic, extremely encouraging, and that's using our standard protocol. So I think that what we know about storage of memory, and here I am specifically talking about long-term memory, not short-term memory that's something like 10, 15, 20 minutes, whatever it is, basically the stuff that you lose if you have general anesthesia, that's not going to be preserved, but that's pretty minimal, and hopefully you won't even be conscious for those last few minutes because they're probably not much fun. But anything else is preserved in structures in the brain, not just the connectome, but the neural body itself. And there's pretty good evidence that we have right now that under decent conditions, again, not guaranteed conditions, we are actually preserving that. So I think that's the way to go right now for anyone who is not very young and very assured of not having a car accident or catching a horrible disease or something. I think cryopreservation is the way to go now, possibly with chemical preservation in the future. Don't kind of dismiss that as, well, I'm not going to need it because Singularity is going to solve this problem for me next year, because I'm telling you that's probably not going to happen. That's not a good bet. So you need to preserve your options. So if we do this, if you get cryopreserved, or if you do make it to a time when we can actually upload you, and I don't know, what the other people here are thinking for the time frame. I have no idea really, but I don't think it's going to be for at least several decades. So if you get cryopreserved or you survive long enough to get scanned and uploaded, what is that going to be like? Well, I'm going to be pretty brief on this because the answer is we don't really know. But I think what we should be aware of is that survival will mean something a little bit different than what it means today, in the sense that if psychological continuity, connectedness and continuity are what make us who we are over time, those kind of connections are going to be alterable more quickly in the future. If we actually are running on a different kind of platform, maybe we can re-engineer that more easily than we can the brain. Not that the brain can't be re-engineered. I think we need to do that. We need to do something about our lousy emotional system, for instance, that our cognitive emotional systems don't work too well together, not that well integrated. And hopefully we can do a better job in the future on a different platform. So I think we're going to have to get used to much faster changes if we choose to make those changes to our cognitive architecture, changes to our motivations. I personally would rather like to better tweak the different basic traits of personality. If you take like the Big 5 personality model, it might be nice if you could change some of those personality traits. So for instance, if you're on a work binge, or you need to have a work binge, maybe you can up your conscientiousness and lower your pleasure seeking for that period of time. That would be kind of nice, and to modify them in different directions at other times. That's one thing I like very much in the very first novel by Greg Egan, this Australian science fiction writer, Quarantine, in which he talked about the power of neurotechnology was not really creating exotic new states. It was the ability to focus, to actually narrow the mind down for specific purposes. I think exotic new states could also be a lot of fun, but I think there is a lot of power in that kind of focus. And that's something that's kind of hard to do with our current architecture. You can do it to a certain degree, obviously, with meditation and just a lot of practice, but there are limits to that. So I think that on a new platform, we're going to become strange pretty quickly. I think we'll kind of change ourselves in various ways. It's not too clear whether we'll all make the same changes, to what extent, or we'll break off into different kind of tribes or clades, as other people have called them. But I think the general trend will be towards much deeper and broader awareness. So there won't be just one kind of awareness or consciousness. It won't be just human consciousness, because it will be detached from kind of the DNA based structure of cognition, and we'll have more opportunities available. So I think we'll increase our sensory input that we link to satellites and environmental monitors and many other things in ways that we can kind of do it by proxy now, but not directly. And much deeper forms of communication may be possible. But exactly what that will mean is probably best left up to science fiction writers for now, because we don't really know at this stage. We're too crude. But the basic point is that we are essentially physical beings, but we're personality and personal survival is an abstraction from that physical structure. It's not identical with it, but it's not dualistic either. We're not some kind of mental, spiritual beings floating around without any physical being. So yes, we can survive either in our own bodies that are reactivated and brought back to function, or potentially by being scanned and uploaded into a different substrate. And there are practical means of actually acting on that today. And then, looking forward to a future in which we have many more opportunities available. So I just kind of wanted to give a bit of a philosophical background for the technical work going on here and point out some of the practical implications of that. But I'm going to quit at that point and maybe take 1 or 2 questions. Thank you.
Thank you very much, Max, and thanks for stopping exactly on time. I think we have the time for 1 question, and if you look in the chat, there was one from Gunther.
I have 15 new questions to look at. So.
They are not all questions. You had this one. Would records of neuron firing in a person's brain, say for 1 hour, constitute a portion of consciousness preservation?
I don't see the question, but if I'm understanding it, recordings of neurons firing.
For 1 hour, would that be useful for something or not?
I'm not really sure. I think probably not, because I think the actual activity over a period of time, I'm not sure what that would tell you. I think, again, given that we know that short-term memories are basically a result of electrochemical activity, and long-term memories are actual physical changes in the brain, I think the physical changes are more important. For instance, I think we don't have to worry too much about the level of neurotransmitters. You could kind of wash those out and you're still maintaining the structure that produced those neurotransmitters and the experience that results from them. So unless I'm misunderstanding, I think probably no, that wouldn't really be necessary. It might be helpful. Maybe it might be something that gives you some parameters to help you reconstruct any missing information, but probably not critical.
Okay, thank you very much, Max. And next is Ken Hayworth.
Hello, thanks so much for inviting me to this colloquium. Let me put up my talk. So this is not a particularly well organized talk, and I will try to be brief so that I can really get to questions, because I think that that's what I'd rather have an interaction with people. But essentially, the main thing that I want to put forward here, and I guess get people's thoughts on, is that I think that the neuroscience community should develop an ultrastructure quality human brain bank, which stores brains in a manner that's compatible with today's best ultrastructure and molecular imaging techniques. And I think that is, I would put forward the controversial statement that is the logical way that brain preservation is going to impact the numbers of people that I think we would like to have impacted by this, and not be just a perpetually, decade after decade, a fringe side path that is painfully slow in its development. I'm very glad to hear that that Max was saying that the unnamed researcher might have first name of Greg, that he is going to be publishing. Let me put forward that if there is any desire to have me or other neuroscientists look at those images pre-publication, I would be ecstatic to look at those. I am still extremely skeptical, but I am always extremely hopeful. But with that, until we actually see the standard cryonics methods actually preserving the connectivity and the ultrastructure of the brain, I simply have to say I don't believe it, and say at the same time that the neuroscience community has been preserving brains at the necessary level for decades and decades and decades. So it's a little bit of a massive disconnect where, you know, gee, for decades the cryonics community has been trying to get to a level where the neuroscientists have been at for decades. Again, I'm gonna try to make the talk short so we can hash it out later. So my take here is that I've suggested to the NIH, of all people, that they should essentially create an ultrastructure quality human brain bank. The NIH has brain banks already. They are very low quality because the quality of those brain banks is set by what is necessary for the types of histochemical and crude anatomical studies that they do on human brains, which is pretty pathetic nowadays. But if you look at what type of research is done on animal brains, it is fantastic, and Randal really put forward quite a few stuff on that. Let me before I go off on this though, I want to say some things that I think that we have in common on this meeting. It seems like we all believe that humanity is likely to continue to progress scientifically and technologically and morally into the far future. It seems like we all believe that the overwhelming scientific evidence that we're simply evolved biological machines, that our brains evolved merely as computational control devices in the service of survival goals, and that our consciousness and sense of self must therefore also arise from these same neuronal computations. And I think Randal put it really well when he was saying that essentially consciousness is a process, that if you're asking the question, what state of the brain is causing this subjective experience, you've already lost. The question is, what process in the brain is modeling that subjective experience? That is the better way. And once you flip that, you are able to see that that a computational process is the explanation of that. In any case, it seems like most of the people on the call here would agree that we are essentially computational beings. We believe that humanity will ultimately, perhaps centuries from now, no time soon, succeed in its quest to understand exactly how the mind and consciousness arises from these neuronal computations. And we believe that humanity will, with this full understanding of the brain, centuries from now, will eventually develop mind uploading, the mapping of a particular mind and simulating that brain And because, as Max pointed out, because we identify as computational beings, we should accept that a simulated copy of that person's mind should count for them as survival. And I don't think I will. I think Max did a better job of that. So I will leave it at that. But beyond that general optimism for a wonderful future that our great grandkids will experience, because they will have the opportunity to be uploaded. Some of us here also believe that we personally, as well as our friends and family members, and frankly everyone else alive today, should they do have a chance at getting to that future through not through medical technology. I agree with Max in this sense. The slow pace of medical progress towards the big problems of aging and a whole multitude of diseases argues to me that brain preservation is really the only option. And so if we can preserve the delicate ultrastructure and molecular features of the brain that encode our personality and our long-term memories, if we can preserve these before they're destroyed by disease and decay, if we can store these for centuries, then we can get to that mind uploading future. And so what I am arguing, though, is that there is only one group of people that really can pull that off. And I don't think it's the cryonicists, and I don't think it's Robert. Sorry, Robert. I don't think that it's any small group of people operating outside of the system. I think it is the neuroscience community as a whole. And so it's my sincere belief that nobody can do this except the neuroscience community, some fraction of it that actually buys into this concept. And I believe that they have the moral obligation to do this. And I realize that is it is not something that is there are plenty of neuroscientists that run away from this like crazy. People are saying great things about Anil Seth. I just finished his book. Last chapter was basically well, I had this I wrote this great book about this theory of the mind, of consciousness. Do I believe it? No, I don't really believe it, because if it doesn't go all the way down to the mitochondria, then it wouldn't really be conscious. And by the way, people that believe in mind uploading are crazy, and death is good. That was pretty much what I got out of the last chapter. So there is a real anger within the neuroscience community for this, and I realize that completely. But they are the people that have the expertise to pull this off. We need to convince them that they have a moral obligation, that this is logical, that this actually is the natural extension of their field. So what do I mean by this? Okay, so the neuroscience community preserves brains by aldehyde fixation all day long. Randall showed some great images. Here's some images that I've taken that show what an aldehyde fixed brain, the resolution that you can get. You can get every single synaptic vesicle, every synapse in great detail. There are groups like this one here that has mapped 100 by 100 by 100 cubic microns of mouse cortex tissue, and mapped out all of the synaptic connections in that, and has seen the signature of learning in that. Randall already showed some of this. This is the great cubic millimeter work from the Microns project, Allen Institute, and Princeton, et cetera. This is the level of detail that the neuroscience community routinely preserves brains at. You know, I want to see brains that are preserved at this level of detail to take any type of claim of brain preservation seriously. And all of this is because you're using aldehyde fixatives. This is another one. This is a human brain that was done cubic millimeter, again with immersion aldehyde fixation from a small biopsy sample from an epileptic patient. We can ask, does the information get destroyed in some way because of the aldehyde fixation? Well, here is a paper that showed that imaged dendritic spines on neurons in the living animal, in a living mouse, and then went and did the preserved the brain with glutaraldehyde, did the electron microscopy on the exact same synapses, and compared the two, and compared the sizes, the relative sizes of the synaptic contacts, and got a very good correlation. Here's another paper that looks into that same type of thing, where they are saying, let's take the best method that we can use to preserve a very small piece of the brain. So if you take a 200 micron thick slab of brain tissue and just flash freeze it, you get something that's closest to the living state. And they did that, and that's what these bottom images show here. And they compared that directly to similar brains that were put through glutaraldehyde fixation. And the result was that they showed that whereas there is morphological changes because of the extracellular space being lost in the aldehyde fixation, the synaptic sizes appear to be the same. And why does synaptic size matter? Well, synaptic size, this is a paper that was recently put out that went and did electrophysiology on pairs of neurons and measured the physiological strength of synaptic connections between these two neurons, and then painstakingly went in and found all of the places where they made synaptic contacts, and was able to add up the area of synaptic connection between those, and ask the question, does the size that you see in electron microscopy of those synapses, does it correlate well with the actual physiological strength? And the answer is yes, they did. They say we find a linear relationship between the synapse size and strength, providing a missing link in assigning physiological weights to synapses reconstructed from electron microscopy. Aldehyde fixation doesn't just preserve the structure of the brain, it preserves all of the proteins. That's what it does. It cross-links them together. And so, papers like this one here that used glutaraldehyde fixation go in, and by various techniques, they are able to do immunostaining for a enormous range of different biomolecules, glutamate receptors and ion channels, and all sorts of things that would be potentially important. I think we should imagine what would a future technology that did uploading on a preserved brain, what would it look like? Well, given what we know today, I think the most reasonable assumption is that it would look like figuring out all of the synaptic connectivity of the brain, figuring out all the dendritic morphology of each and every cell, so that you could create the structure of a compartmental model, each of the neurons, looking at all of these synaptic connections and their sizes and associating a physiological strength of that synaptic connection. But then you have things that may vary that are not seen in electron micrographs. There might be ion channels that are ion channel distributions in neurons that are not able to be assumed from prior knowledge of the morphology. And so what you would want to do is you would want to identify those particular things that you can't fill in with parameters and develop markers for those, which means that you might have a subset of proteins that you need to label and image. You might have a subset of the messenger RNA expression that you need to look at to see what particular proteins are being expressed in this neuron versus this neuron in order to get those parameters. Well, the aldehyde fixation is preserving all of that. Here is a paper that goes through just an enormous range of antibodies that can be applied to fixed tissue that not only finds the particular proteins, but is specific to the phosphorylation states of those proteins. Here's a paper that measures the ion channel distribution on particular dendrites, again, in fixed tissue. Here's a paper that shows that glutaraldehyde fixation can be quite compatible with messenger RNA assays. And finally, here's a paper that used super resolution STED microscopy on not glutaraldehyde fixed tissue. This was actually glyoxal fixed tissue, but it's similar. It's fixed. That went through particular synapses and used antibody labels to label particular proteins. And they labeled all of these proteins in different. They couldn't do it simultaneously, of course, but they could look at this particular protein in this synapse, and then on a different piece of tissue, this particular protein on this synapse. And this shows the coverage of aldehyde fixation for preserving the information content of the brain. All of this information about the quantities was there and proven by these neuroscience papers. And whereas a lot of these things are small scale studies, there's things that are happening today, like this paper here, where they sliced up a whole rhesus monkey brain into 300 micron slabs, cleared each of those slabs and imaged a particular set of proteins, in this case, to do tracing of fibers across the entire monkey brain. This is the kind of routine stuff that is happening in the neuroscience community today. So, you know, let me just. The neuroscience community could have before this said, yes, we can do all of these things. And yes, of course, we're committed to the idea that the brain is where personality and memory and everything is at, but we can't preserve those things. We can do these assays, but we can't show that we can preserve something for hundreds of years into the future. And that's where Robert really, his technique, showed that could be done. That was the missing piece that he showed. I'm sure he's going to talk more about this, but the idea was, he said, look, these aldehyde stabilized preserved brains, they are robust enough so that you can perfuse them at room temperature with cryoprotectant and to a level that is necessary for true ice-free vitrification. And he published it in this paper. Here's a rabbit brain that is vitrified solid, had been at minus 130 degrees Celsius. We did electron micrograph images of that. Here is that brain that rabbit brain that had been vitrified solid at minus 130 degrees Celsius. Any neuroscientist looking at this knows immediately that the connectivity is preserved. They have no question in their mind about it. It's not a, gee, we don't really know, but it might. No, it is. And I showed that for a pig brain. And so I just want to end with this idea of the neuroscience community. In order to get the neuroscience community to embrace brain preservation, I think the way to do it might be to simply say this. You already have brain banks, but you have brain banks for the crappy imaging technology that you do. You have human brain banks for the crappy imaging technology that was like 30, 40 years ago. Imagine a human brain bank that preserves intact human brains that are compatible with all of the fantastic crap that you're doing today, including the highest resolution electron microscopy, the super resolution light fluorescence microscopy that is looking at all sorts of different proteins. Find a method for brain preservation or a brain preservation bank that allows whole human brains to be compatible and stored for an indefinite amount of time that are compatible with those imaging techniques. That is the way to get this into everybody's hands. And I would argue that the neuroscience community has they could make this easy. They could make this free, okay, free to everyone in humanity. They could change the laws so that this was part of a death with dignity. That brain donation was part of death with dignity. They have the institutional stability. They have the surgical knowledge. They have everything that needs to be in place to get this done very quickly and reliably. Let's ask ourselves why we're not concentrating on making that happen, and we're trying to twiddle over here outside of the mainstream science and neuroscience community to get a much worse product. That's my talk.
Thank you very much, Ken. We have time for a couple of questions, perhaps, but I have an observation first. Coming to the point that the neuroscience community has a moral obligation to do this, and is also the best, the only qualified actor that can do this. I would agree with this in principle, but I remember that very many years ago, I remember you saying exactly the same thing, and I don't really see too many indications that things are about to change. Now, very pessimistically, I'm afraid, I think this is just not going to happen. You're not going to get the mainstream establishment neuroscience community on board on that. And that, that can be discussed, of course, but that is how I see things at this moment. In view of that, I very much disagree with you, and I think the right actor that can get started is not the mainstream neuroscience community, but perhaps some very small part located on the extreme fringe of this community, because they are going to be the only one who want to get this done.
So first of all, I want to say that I completely realize that I remember people at conversations with the cryonics community 12 years ago, where people said, look, even if a prize was won that showed brain preservation was possible, nobody would care within the scientific community. And they were absolutely right. Fine, 100%, at some level, I completely agree with you, but this is the hill to die on. This is the hill to die on, for me at least. And I am going to keep pushing the neuroscience community to point out that this is the logical consequence of a successful neuroscience, that mind uploading is the logical consequence of a successful neuroscience, and that the neuroscience community has this moral obligation. And I completely agree that that most people that it's probably right that this will never work, but it doesn't matter to me, because this is the right way of doing it. It's the right way of doing it. Does that make sense? Probably not.
It does. I still disagree, but it does make sense. Let's choose a couple of questions in the chat. There is one from Gunther. The question is, there is a voltage potential required for the synapse to send impulse across. Is a mechanism of generation of this potential part of our consciousness that needs to be preserved?
So there are neuroscience models of how neurons interact with each other. Those neuroscience models are very clear and answer that particular question. And so the question really becomes, can you extract from a brain enough parameters to make one of those simulations? And I think the answer is yes, but getting into the details would be would take quite some time. I understand that that there is a question of, do you get parameters from structure versus do you get parameters from voltage recordings, etc. Those are two different methods, but I think the structural way is the only way that we really have to go right now, and I think that is viable. But maybe we can talk later about that.
There is another question from Gunther, and let's hit his last question and make it quick. Gunther asks, it is a common practice to preserve brains from donors in formal data. Would, in your opinion, aldehyde preservation be in favor over the cryoprotectant filling vitrification and lowering the temperature down to minus 130 Celsius?
I'm not sure if I got the full question.
If you scroll up to minute 1752.
Let me answer it. Yeah, let me answer it this way. I think that there are many possible routes to brain preservation. What I would love to see is that the neuroscience community explore those routes. There are routes that involve low temperatures. There are routes that don't involve low temperatures. They should both be explored. There's routes that, like cryonics, that explore not using aldehyde fixation. Absolutely, that should be explored. Tons of research dollars should go into it. All of these avenues should be explored. I think that given what I've seen, aldehyde fixation is overwhelmingly used in the neuroscience literature to study these very things that make us us. And so it obviously has the pole position on being the best technique.
Thank you very much, Ken. And let's now give the floor to Martine Rothblatt, who I see has a very nice Zoom background.
Thank you. Thank you, Giulio. Let me try to share my screen here. Full screen. Yes, I thought, Giulio, might have to ask you to advance the slides for me, if that's okay.
Yes, I can do that. The only problem is that if we do that, the others will not always see
Okay, let's see if I can.
your screen in the front. So let's do that as last resort. Now you push share screen at the bottom of the Zoom screen.
Yes.
Then you have a choice of windows from which you can choose which one you want to share.
Yes.
In the meantime, I'm going to set up your
Unfortunately, the security on this computer doesn't like me to share this Zoom screen.
okay, so let's do this and one little moment. That's a real-time experiment that we are doing. Let's see how this
I think
works.
I might have cracked it. I might have cracked it. Yeah.
Okay, so let me stop this. Let me stop this. You try, and if it does, yeah, I see.
Yeah, I think I have to exit out of Zoom in order to make this change, which I don't really want to do.
Okay, so let me do that for you, and you ask for the next view when you want it.
Beautiful. Thank you so much, Giulio. So I've really enjoyed listening to everybody, and thank you, Giulio, so much for organizing such a great colloquium. If you could move to the next slide. I'd like to talk about the LifeNaut approach to creating cyber consciousness. This text slide reviews the fact that LifeNaut is a public-facing online research program. It's operated at no cost to the user by a not-for-profit foundation. It's the Terasem Foundation. The LifeNaut.com platform is a hybrid of customized software and open source software designed to create a quote-unquote mind file of a user for future activation into cyber consciousness via future developed quote-unquote mindware. The software is frequently updated and advanced, and we're always trying to actually tilt things more and more toward ultimately completely open sourced. What we call a mind file is just a word we created to describe an organized database of a user's mannerisms, personality attributes, memories, feelings, beliefs, attitudes, and values. as reflected in uploaded and tagged videos, images, documents, journal entries, lists, psychological tests, Bainbridge opinion inventories. Those are huge two-dimensional opinion assessments by this sociologist, Bill Bainbridge, who's created with some degree of validation over 100,000 questions with a chance to do a two-dimensional Likert type of opinion on these 100,000 plus questions, and with some type of algorithmic analysis that aims to draw conclusions. And finally, just plain old-fashioned chatbot training. You can train a chatbot to be more like yourself. So that's what we mean by the word mind file. By mindware, we simply mean some kind of a consciousness operating system that would convert a mind file into an interactive approximate analog of its source mind. We have quite a few people participating in this experiment. There are now over 50,000 mind files established at LifeNot. And more all the time. Some are, most of those I would describe as being inactive. People tried it and stopped. There's something like maybe a couple thousand that are really active, and I think in that sense more useful. Our philosophy behind this LifeNot experiment is really a philosophy born of Teilhard de Chardin, which is that consciousness is sort of a collaboration. We all realize who we are. We have a sense of who we are. We have a constant identity of me-ness. But that me-ness is also a collaboration with everything that we've interacted with, people, media, the environment. And all of that environment is in turn a consequence of historical environments. So consciousness is a collaborative process. That's basically our mindset. So that even though trying to get your mind into a database that is structured by a lot of other people, to us is not really inconsistent with it being a reflection from which consciousness can be gleaned. The purpose of this LifeNot project is to test an open Turing type of hypothesis. The hypothesis being tested is that a mindware activated mind file produces a consciousness that is functionally equivalent to that of the source mind. Now, users of LifeNot can elect to opt out of participation in testing the Terasem hypothesis. Actually, this slide is not really written quite correctly. They actually have to opt in to participate in testing of this Terasem hypothesis. And whether or not they opt into the test, they can still use the mind file features of LifeNot as some sort of a digital memory bank for their friends and family and posterity and whatnot. So as noted, they have to provide affirmative consent to have their mind files activated or attempted to be activated with future mindware. And the null hypothesis that we're testing, the lack of equivalence, would be, according to the terms of this experiment, would be disproved if a panel of appropriately credentialed psychologists deem, after a more or less continuous interaction with the activated mind file over a period of a year, that the analog mind is equivalent, in their opinion, to stored evidence of the source mind. So we have not yet impaneled such a psychological team, and we're really wide open to every sort of opinion for ways to test whether or not the purported consciousness that we would reactivate would in fact be equivalent to stored evidence of the source mind. I think, a really good artistic depiction of what we're trying to do is this Alex Gray drawing, where we really are not kind of debating the question of whether or not the reactivated or activated consciousness is precisely the same as the source mind. In fact, we are really very much in tune with what Max Moore said in his presentation earlier today, that, he himself and in his well-researched dissertation demonstrates that, consciousness is a completely transitional state, and that he's not the same as he was yesterday, the year before, nor is any consciousness. So instead, what we're trying to do is to create a functional equivalent. And whether or not that means that the functional equivalent feels that they are a different person than the stored original, personally, I think they certainly will. That's really not the point of the experiment. Again, the point of the experiment is to have experts in mind science, psychologists, in other words, in mental health, deem that, in their opinion, based on the evidence that they have, that this digital functional equivalent, purported functional equivalent, is in fact functionally equivalent to the original consciousness, even if not exactly the same in every regard. And people are excited about participating in the experiment because they generally view it either fun to participate in this experiment. They believe that they'll actually prove the null hypothesis, that there's a lack of equivalence, or they're of the view that will disprove the null hypothesis, that there will be equivalence, and that they would feel that they had continued their life in this digitally functionally equivalent frame of consciousness of themselves. They might feel they've continued their life in the way that a child continues their parent's life, or a friend continues their friend's life after the friend's biological body disappeared. Or they may simply feel that, in fact, they are themselves, they're the core of themselves. They're not every aspect of themselves. They miss their body, or maybe they don't miss their body, but in some way they feel they are a continuation of themselves. And that's what I think Alex Gray does a great job of depicting in this piece of art. There are a whole lot of ethical aspects of cyber consciousness, and I discussed them at length in my article in the Journal of Machine Intelligence, I believe it is. But basically, we take the ethical aspects seriously. As I mentioned, you have to opt in to participate in it at all. But perhaps what we are most concerned with is that we are not bringing into being purportedly or possibly cyber-conscious states that are anxiety-producing or not wanted by the nascent cyber-consciousness that would be brought into being by mindware being developed throughout the world right now. So first of all, the LifeNaut project includes assisting any consciousness as psychological assessors advises exists with living a life in accordance with their expressed autonomy, subject to the socio-technological constraints that we have. And those may be resource constraints, just any type of social or technological constraint we have. if the revived consciousness wants, for example, a perfect nanobiotechnological body, we're probably not going to be able to provide that on day one. If they want more interesting things to do in the metaverse, that's probably something that we could help with. Secondly is the point that I mentioned at the beginning of this slide. We want to impanel an IRB to approve protocols to be put in place that would respect the autonomy of cyber consciousness to the extent practical. The project expects that there's not going to be a kind of a digital either or on off state of cyber consciousness, that first there's none, there's just this dead mind file. And then with the flip of some consciousness operating software, or aka mindware, suddenly there is a a perfect reflection of the original source mind consciousness. Instead, we really expect that aspects of consciousness will be able to be brought into being bit by bit. And we need to figure out ways to respect the autonomy that is expressed in those aspects of consciousness, short of full human consciousness and short of the full human source consciousness. So again, this article that you could find online goes into depth on the type of protocols that would be deployed to basically kind of bubble off sections of possible consciousness, so that each section could be assessed and interacted with and determine to what extent autonomy is present before we expand the bubble of consciousness operating software to greater and greater extents of the mind file. To allow some time for questions, let me wrap up with a huge thank you to Marvin Minsky. Our entire project is really premised on the thoughts and teachings of Marvin Minsky, starting with Society of Mind, and perhaps most empowered by The Emotion Machine, where he lays out pretty clearly a kind of a structural pathway for creating a cybernetic reflection of human emotions, which he feels are very much at the heart of human consciousness. So thank you, Marvin. We miss you. We hope to see you back. So Julio, with those introductory slides, I'm happy to start the discussion period until you say the time's up.
Thank you very much, Martine. Let's go have a look in the chat if you have some recent question. Right, we have one from Michael Perry, who says, I understand that you also preserve a genomic record for people that desire it. It seems to me that that would be an important addition to your Mindfile information, and the people who choose to submit a Mindfile ought to routinely choose this relatively inexpensive option also.
Yeah, it's a certainly a valid point, Mike. So everybody's on the same page with Mike. The LifeNaut project includes an option that I believe for $99 you can store a sample of your DNA in liquid nitrogen under kind of good clinical practice protocols, so that it's safe and well validated. Not very many people have chosen this option. I think we have maybe about 30 or so people who have, although certainly other people have stored their DNA other places, and there's no shortage of places to store one's DNA. I personally, and here I'm just giving my own opinion as a presenter at this colloquium, I'm personally fairly skeptical about the contribution of that genomic material and about the utility of it. Nevertheless, I certainly respect the opinion that some aspect of your connectome is hardwired in your genome, and that portion of your connectome that's hardwired in your genome could perhaps be of assistance to a consciousness operating system in trying to make a sensible analog of your mind out of the amount of information that is stored in your mind file. But I will say that in just my personal opinion, while I respect that point of view, it's not really my point of view. My point of view is that if you have an adequately robust mind file with enough reflections of your mannerisms, personality, memories, feelings, beliefs, attitudes, and values, using all of the dozens of different tests, psychological tests and Bainbridge modules and whatnot that we have on site, that I'm a believer in the fact that at some point in the future, consciousness operating software is going to be developed that would be able to resurrect, if you will, a self-aware, subjective consciousness feeling analog of yourself that would realize exactly what it was and exactly what it wasn't. And if we do a good job, would be very happy to be reactivated and able to interact in the world.
Thank you very much. We have a question from Robert McIntyre, who says, I like the IRB, Institutional Review Board, protections for uploaded mind. Do you have a draft or other document to share? And he says, I have taken some steps at this myself in the past.
So I really welcome the help in developing this IRB. We have not yet created it. It's something that we need to do. I was really looking forward to participating in this colloquium so that I would hope that we could get some additional people to join our effort in creating this IRB. There is a stab at this IRB in the Journal of Machine Intelligence. It could be Journal of Machine Consciousness. Sorry, I don't remember the name of the journal, but I'll look it up and I'll put it in the chat field. I'll put the link to the article because you can get it online. So I do make a kind of a stab at what should be in the charter of the IRB, but it really is a first stab. But I look forward to the help of everybody and making it like a bulletproof, really good IRB.
We have a question from Natasha. How has MindFile changed over the past decade? What new technology and security is offered?
Yeah, so we have made a lot of improvements. Thank you, Natasha, for the question. We've brought on board, we used to contract out almost all of the effort. So we've brought in-house to the LifeNaut project expert software engineers who are constantly working to give us state-of-the-art security protection. We've distributed the mind files across across 3 separate locations, one of which is completely offline, disconnected from the internet, written on what's called millennial drives. These are drives purportedly will maintain the validity of their information for 1,000 years. Hopefully it won't take that long to get the mindware up and operating. So yeah, we're worried about that, Natasha, and I think it keeps the staff up at night they view themselves as having, sorry to import a kind of metaphysical concept here, but they view themselves as having a sacred obligation to protect these mind files. They feel that they are lives, that there's at least life imminent in the mind file itself, that for many of them have expressed a desire to have that imminent consciousness re-expressed. So the staff takes it seriously. And our point of view is that software, firmware, security protocols constantly change. And so that we have to constantly keep up with it. For example, we store offline at least every week. We make a fresh storage offline so that we're always up with all latest storage protocols and media. So it's a living database in every regard, and our security protocols are living as well.
Thank you very much. I see that there are a few people in the audience who want to sign up and want to know how. I think they just go to lifenow.com. And Robert, I don't quite understand your last question to Martine. Now, if you can just switch your microphone on since you're the next speaker and ask your question yourself.
Sure. So I have two questions. One is you described earlier that the null hypothesis for testing whether this idea of the mind file is successful or not would be you sort of record someone while they're alive, you gather some amount of data, and then after that person has died later, once you've developed the software, you'd then put the data in, and you'd have a team of psychologists essentially evaluate if, in their opinion, more or less the mind that's generated matches the data. And it seems to me that the right way to test that would instead be to take a contemporary person in the future after you develop the mind file, extract the data, over a year, however long it takes, and then you compare that the still living person with the generated person by a panel that includes that person's still living family and friends and boss, et cetera. So I was curious what your thoughts would be for that, and then I have one other question after that.
Yeah, thanks, Robert. I totally agree with you that that's a better experiment. It's like no doubt about it. It's a better experiment. The reason we are doing this experiment is actually to kind of kill a couple birds with one stone. So there is if I'll say it, the subterranean bird that we're trying to kill with this stone is the desire for some type of techno-immortality on the part of the participants in the experiment. Somebody like myself who is a member of Alcor, and I do place definitely 95% plus of my confidence in terms of a reasonable route towards some kind of technological life extension in Alcor. But in the worst case scenario, like everything in Scottsdale disappears, this is like, the next fallback after Alcor. So by participating in this, individuals have a chance to kind of give some muscle, if you will, to their desire to have continuous life extension, even beyond what Alcor makes available. On the other hand, it's not really that interesting for some of us to do this purely for life extension purposes. We want to participate in something that is more scientific in nature. And even though it's a long-lasting experiment, and even though I do think that the experimental parameters are not as good as the ones you described, Robert, because looking back at like hundreds of hours of video with somebody is just not the same thing as interacting with the person, live in some sense. It's basically the it's the best experiment we can make out of people's desire to have some pathway to techno-immortality. I think that would be like the straightest up response to your question.
Gotcha. And then, if I was going to make what I would say is the most powerful argument against this this type of procedure, which essentially is have some sort of structured information gathering from a person, like a super in-depth interview, essentially that takes a long time while they're alive, and then attempt to recreate them. I'd refer to essentially hidden bits of information in a brain that either cannot be compressed into language or just aren't ever compressed into language. And so the example I'd use is, right now I have a 120-bit password in my head that opens my computer. And I would argue that it's simply impossible for you to ever infer that from any type of personality quiz, regardless of how many years you take to do it. And if something so basic like that can't be extracted, it seems like the whole thing is hopeless. Like, it would actually convince me. I'm trying to think what evidence would convince me that this has a hope of working to properly reconstruct someone. If you could get that 120-bit password anyway, where it's like, yeah, after you ask someone enough questions, like, that's just a thing that kind of falls out of it, I would believe this works. But it seems clearly true that you can't do that. And so it feels like that any sort of personality quiz is essentially the equivalent of trying to preserve like a Blu-ray DVD by simply snapping a picture of it with your cell phone camera. Like, it's just not going to get enough data. So I wonder, do you believe that you could get that password with enough questions, or is it kind of a more subtle thing where you think that all of the hidden information, like the password, coincidentally is irrelevant for maintaining someone's personal identity?
Yeah, I don't think that there's going to be any way that with the tools that we have on Life Not Today, that we're going to be able to get a snapshot of every thought inside of a person's mind, or even every pathway of thinking inside of their mind. In fact, far from it. I would, if I had to just come up with a rough guesstimate, I'd say we'd be doing great to get like 10%. Okay. So, no, I don't think that's possible today. Do I think that, there are Neuralink-like efforts that could ultimately, ferret out that information, neuro nanobots, et cetera? Sure. I think, all of that stuff is technologically possible. So I do think there would be, I don't think it's beyond technology to get that 100-bit password out of your head. But we're not able to do it today. The Life Not project would certainly be open to including all additional ways to capture what we call mind file data. So the project is funded to be open to any novel ways to get the substance really of who we are and our thoughts and our more subtle information out. So yes, I definitely would like to have that. Now, your ultimate question is, do we end up after, let's say, this mindware is developed, do we end up with a snapshot of the Blu-ray? I'm not sure that would be the right analogy. I'm not confident that, on the spot right now, I could come up with exactly what the right analogy would be, but a rough guess would be maybe we would have the soundtrack of the Blu-ray, but not all the amazing imagery of that, 2-hour movie. And is the soundtrack the same as the movie? No. Is the soundtrack the guts of the movie? Kind of, yeah. Kind of, yeah, depending on kind of how good the soundtrack is, and depending also to what extent the soundtrack tracks to the movie. One of my favorite movies, this movie about the first dog named Alpha, has virtually no soundtrack at all except growling. I don't think that you can, really get the movie from the soundtrack. On the other hand, there are other movies that's just dialogue the whole way, and you probably don't even need to watch the movie to get the idea. So I would say if I ended up 100 years from now with a soundtrack of my life, without all the visuals, I would feel pretty happy. I would feel like, whoa, this is absolutely astonishing. I kind of, hyper jumped into the future 100 years from now. And I really don't remember what my houses look like, but, I remember that I lived in this house, I lived in that house, and so that kind of thing.
Well, and you can't deny that it's useful. Like, I mean, as someone who spent a long time, like many hours, like recording family members before they died, like it's amazing how little people care about that beforehand and how much they care about it after the person passes away. So, okay, I think we mostly agree about that then. We just have different kind of terms to describe it.
Well, anyway, let's thank Martine for the talk. And there are, if you look at the chat, Martine, there are some comments, especially from Max and Natasha, about synergies between Alcor and the Terasem Life Note. You might want to read that.
Yes, and also
answer them.
Is that link somebody put in there, the link to the article?
I don't think so.
Okay. I'll put the link to the article in there. Thanks, Julio. Thanks,
Thank
everybody.
you very much, Martine. So, Robert McIntyre, the floor is yours.
Okay. Let's see. Share screen. Is everyone able to see this screen?
Yes, we are.
Cool. Okay. Yeah, so this is going to be a shorter talk, and I hope to be able to open it up to discussion afterwards. And while I'm certainly excited to talk about consciousness afterwards, it doesn't like directly address this issue. My assumption is a very materialistic, very practical one. And so what I'm going to talk today about is what I learned running a human brain bank for how to design an effective memory preservation protocol. So for those of you who, so the overall like pathway of this discussion is kind of broken into 3 parts. So the first part is I'm going to talk about what I think the standards should be for a brain preservation protocol, that if you meet those standards, it's virtually guaranteed to preserve memory, or neuroscience is disastrously wrong about how memory works. And I think that those are the standards that can be met, that they can be defined today, and that given that they can be defined today, it makes sense that they are the standards we should hold preservation protocols to. The second thing I'll talk about is, from running this human brain bank, it seems to me that we have bad news, that the current way we're approaching this preservation is very unlikely to work. And in a somewhat surprising way, I would argue that even if you can immediately preserve someone after death, still, even in most of those cases, it's not going to work to adequately preserve them according to neuroscience standards that we've defined before. And then we'll briefly touch on, okay, so then what can we do about this? Now that we've done this human brain bank and we've learned some useful data from it, where do we go from here? So for those of you who don't know, which maybe is none of you, I'm a researcher that works on brain preservation technology. In 2015, I created this aldehyde stabilized cryopreservation technique that won the Brain Preservation Prize that's run by Ken Hayworth of the Brain Preservation Foundation. And the overall idea behind this preservation technique was to chemically cross-link proteins throughout the entire brain, and then do the cryobiological approach of water displacement through cryoprotection and subsequent vitrification. When you do this, you get virtually the same quality that you get just from the aldehyde fixation. And that level of quality is sufficient to preserve the entire connectome, meaning the connections between the synapses in the brain, that's thought by neuroscience to be sufficient for preserving memory. So that's just a brief background of where I'm at. Since then, I've started a company called Nectome, and I'm working to develop this preservation technology from the very pure controlled laboratory environment. And I'm seeing, what does it actually take to get these same quality results, but with human brains. So that's what I've been working on over the past several years. And then, you know, over the last 5 years, or I guess 4 years, but specifically the last year and a half to 2 years, I've been running a human brain bank in Washington, Oregon, California area that covers about 30 or so hospices that are participating in this program. And you know, this is, as far as I understand it, the first effort to analyze cryonics protocols through the methodology of an actual brain bank. So these people donate their brains to this brain bank. There's no expectation of long-term storage or preservation. We apply preservation protocols, and then the endpoint is that we take those brains and slice them and analyze them, you know, identically to how we would for the Brain Preservation Prize. These are some images from the brain bank that are light microscopy images that look, you know, I would argue better than most other brain banks. And this just gives a little bit of the flavor of what we've been able to do. This brain bank is radically different from most other types of brain banks in that it's a rapid response brain bank. So, you know, the traditional way that brain banking might happen at a group like the Harvard Brain Bank, for example, is someone signs up to donate their brain either before they've died, they sign up themselves, or after they've died, their family can then donate their brain. And then they're generally held at a morgue, you know, presumably they die in a hospice or a hospital. They go to a morgue, they're held there for a day or two. Eventually, there's a chain of custody that's generally 3 or 4 different organizations, but a representative from the university or someone that's contracted by them will eventually come to where the individual is, and they'll remove the brain and then they'll do immersion fixation. Often the brain's cut into the 2 hemispheres, and one of them is frozen, and then the other one is immerse fixed, although sometimes the entire brain is fixed. And because the progression of the wave of fixation proceeds about 1 millimeter an hour, it can take a very, very long time, several days to even a week, to actually fully fix a brain with immersion. And when you add up the long amount of time, you know, pre-mortem, post-mortem, these brains don't look particularly good. It's very rare in a brain banking scenario traditionally to have a brain that's less than 4 hours post-mortem. It's almost unheard of. Average more like is 8, 20, you know, 36 hours, that type of thing. Whereas with my system, we have a lot tighter coordination with the donor and the donor's family, so that we've already set up all the equipment necessary at a local funeral home. And then as soon as that person passes away, we actually have a team that's near their house that's funeral service professionals that go and do the extraction, move the body to the local funeral home, and then we do the preservation as quickly as we can. So the amount of time that has passed from declaration of legal death to the beginning of perfusion is generally, you know, we've had 45 minutes, we've had an hour, 2 hours, 90 minutes. Like that's typically what that looks like. And then since it's done with perfusion, you have very rapid preservation of the entire brain. So that's what I've been doing. And so, you know, I've gotten these brains, we're preserving them just with fixation, right? So we're not even doing the cryoprotective parts of this. We're just looking at what these brains actually look like once they've been preserved. And the way that I'm preserving these is in many cases faster than cases that Alcor has done, but at the very least, it's fairly comparable with the scenario of like a quote unquote natural death, where you're in a hospice, you die, and then people try to rapidly preserve you as quickly as they can. So that's the background. Now, we'll get to the bad news in a bit, but you know, just hold that in your mind that that's kind of my background. That's the brain bank that I'm doing. I'd like to talk about what I think makes sense in terms of what a standard for a cryopreservation procedure should be. And to do this, we should look at what the anatomy of a preservation scenario is. So I think that dividing it into these types of pieces makes sense for a lot of reasons. So typically, in a preservation situation, you have a lot of time that's pre-mortem. So this is before a person has passed away, but they may be having difficulties. They may be in a coma. This period, you could consider it to occupy the entire lifetime of the individual up to their legal death, but in practice, it's the last week or two. So I think that's an important region of time. Then you have the declaration of legal death, which, as we all know, is not necessarily true death, true destruction of information. And then after the declaration of legal death, at least for any protocol that's done in, that has ever been done for preservation, then you have a period of surgery that takes some amount of time to gain access to the vasculature of the individual. And then I would say you have blood washout also that would be part of the surgery. And then there's preservation. So the preservation is the point at which the biology becomes more stable and becomes less liable to be damaged and less time critical. And then you have long-term storage. So I think this is a useful breakdown of the timeline of what a preservation looks like. And in order to develop reasonable standards for preservation, I think that it somewhat cleanly splits into two pieces. So you have this section where the biology is characterized by being very fragile and degrading, that I'm calling the biological part of the preservation protocol. And then you have a section that happens after the preservation is done, where the individual has been converted into an artifact that's stable and it's not as time critical. So I think that when we think about how should the standards of preservation look, what does that document look like? You have different sets of standards for the biological and the artifactual components of this preservation, because they're very different in nature in terms of how time critical that they are. So for the biological section of this, again, it's characterized by increasing damage being done to the brain. You can imagine there's damage that happens before death, there's damage that happens at death, there's damage that happens while you're trying to gain access to the vasculature. And the standard here, I think that makes sense, is that all of this should be clinically survivable, meaning that every step of this procedure, in theory, should be reversible. Again, I'm trying to develop standards where if you do meet these standards, then it means you're preserving memories. So these are standards that are kind of lower bounds for that. And it's not to say that if you don't meet these standards, it's guaranteed that you're not preserving memories. I'm trying to provide standards that if you do meet them, then you can say with high confidence that you are preserving memories. And so clearly, if every step is clinically reversible and clinically survivable without impairment of memories, then almost by definition, you're preserving memories. So think of like a heart attack victim, right? If you suddenly get cardiac arrest and then there's a team that immediately goes and is able to restore your life after like 3 minutes of ischemia, right? Then if your memories are still intact after that, that demonstrates that that part of that protocol at least is reversible and survivable. And so it's very difficult to argue that 3 minutes of ischemia after a sudden cardiac arrest in some way deletes memories, because we have strong clinical evidence that it doesn't. And so therefore that could in theory be part of a preservation protocol. If that was all that happened, sudden cardiac arrest and 3 minutes of ischemia, and then you're converted to an artifact, that would be an example of a protocol where you'd be able to say with high confidence that at least the biological part of it is sound, that it's not damaging memories. The second part of this is the artifactual component. And this is characterized by, you've taken someone who is a living person and now they are an artifact. They're a thing that's not alive anymore. Maybe it has a potential for life later, but if you're a glass statue, it's not participating in the activity of life, whether that's done with fixation or not. And so, and that may not be clinically survivable. I think that we can define standards for a preservation protocol before we have full reversibility. But given that you don't have full reversibility, then what makes sense as a standard? And I think the standard for this is that engrams, memory engrams, as properly understood in the neuroscience community, are preserved according to information theory, as it's commonly understood the information theory community. So what does that mean? What that means is that different mental states that are genuinely different from each other need to become different physical artifacts. If that's the case, then the preservation is injective and information preserving. If it's not the case, then you may not be preserving memories. Again, you might be able to argue that in certain cases, you don't care about the differences. But again, these are standards to show that if they're met, they actually preserve all the memories. So an example of like the mind file stuff we were talking about before, like I have, the hard test for this is consider a version of me that has a slightly different password. Okay, like I have a 120-bit password in my head and it unlocks my computer. So consider a copy of me where it's identical in every respect, except it genuinely has a different long-term memory. And the different long-term memory is that there's one bit that's flipped in that password, just one, so, that would hash out to an ASCII character being changed, right? Those two organisms are remarkably similar. Almost any experimental test you would want to do is not going to be able to distinguish them. In fact, the only real experimental test that's not going to just give you noise if you try to compare them is try to enter your password, right? One of them will faithfully put in password A, one of them faithfully put in password B. That's a long-term behavioral difference. They're going to continue to have that difference over days, weeks, years, et cetera. And so I would say that's like one of the main good comparisons to test whether something is genuinely preserving memory is, could they keep even as subtle a difference as a single bit in a long-term memory being different? Could they keep that distinct? And as we understand in neuroscience, there would have to be some constellation of synapses that are different between those two organisms if they're genuinely going to type different characters on a keyboard to unlock their computer. And so the standard that the Brain Preservation Foundation has created, and that I think is reasonable for this, is if you're preserving the entire connectome, and you're preserving, essentially most of the proteins that exist in the brain, then according to current neuroscience, you'd be able to preserve the type of distinction that exists between organisms that have different long-term memories. So that's what I mean by engrams preserved. You can certainly try to walk this standard back and say, well, what if you have a process that doesn't preserve every single synapse, or doesn't preserve most of the proteins? But again, you're then departing a little bit more from more commonly accepted neuroscience theory. So we have two regions, we have two sets of standards. We have clinical reversibility, we have information preservation. And I think if you can meet both of these sets of rigorous standards, you can say with high confidence that memory is preserved, because there's hardly any wiggle room. You're survivable up to the point that you're turned into an artifact, and the artifacts are such that either they preserve memories according to information theory, or neuroscience is disastrously, catastrophically wrong about how memories work. Which leads us to the bad news here that I've gotten from running this brain bank. And that is that I don't think the current style of cryonics has much of a chance of working reliably, regardless of how well it's done. And the reason for this goes back to our biological standards here. The way cryonics is practiced today, I would call it reactive preservation. The idea is that someone signs up, and then at some later point in their life, they die, hopefully with some advance notice, but perhaps not. And then a team swoops in as quickly as they can to try and do a preservation. And I think this sort of matches our intuition to some degree in critical care medicine. Like, we all know that if someone has a cardiac arrest and they fall down on the ground, that if you're fast enough, you can recover that person. And so clearly there's some window that exists after that person has entered a state where they could be legally declared dead, where you could preserve them. And so clearly the information that's in their brain doesn't just magically disappear the moment their heart stops. And I think that cryonics, as it's currently practiced, this reactive preservation, is built on this intuition that there's always a window, right? That we look at this example of a cardiac arrest and see that there is a window of time where you can recover someone. And then we infer from that anytime legal death is declared, there's still some window that's maybe as long as it is with cardiac arrest, where that person's information still exists. And so as long as you operate within that window, you're likely to be able to preserve them. And I would very strongly challenge that assumption based on what I've learned from this brain bank. The reason is that the way a person dies in a hospice scenario is nothing like a sudden cardiac arrest. And so the idea that there's a window, that analogy that you make from a certain cardiac arrest situation is completely inappropriate in this case. I've learned from personal experience that people do not die on any sort of timely schedule, and that the agonal phase, meaning this period of time where a person's lost consciousness or they're causing major problems with their biology, and the actual declaration of legal death can be very, very extended. To give you an idea, you can have a situation where someone is in a coma for a week, but their heart is still beating. And by the time they're finally declared legal dead, they actually have necrotic tissue in their brain because it's not been perfused well. I mean, almost by definition, if someone is in a coma during the agonal phase, like their brain is not being perfused well, their brain is not getting what it needs to actually function properly, and thus it's accumulating damage. And my argument is that, back to our standard for biological preservation, that the actual agonal phase of dying isn't clinically survivable. There's never been experiments where, because it'd be unethical, where we've taken someone who's gone through 4 hours of an agonal coma and then tried to revive them the way we might try to revive someone who has suffered cardiac arrest. But just based on the fact that you can have severely bad perfusion of a brain, severely bad oxygenation of a brain, and that people actually can develop necrotic tissue while they're in this coma, it seems very clear to me that it's not survivable, that you wouldn't be able to recover from that with your memories intact, that it's radically different from just suddenly having a cardiac arrest. The other problem is that you can have pretty severe blood clotting even like before your heart is stopped, and this can really drastically interfere with your attempts at preservation. Depending on how the person dies, they can have very rapid clotting afterwards. And so my conclusion from this is that in a traditional hospice setting where a person dies from total organ failure, which is a very common way to die, where you have progressive failure of one organ system after another, and the whole time the brain is bathed in toxic byproducts, and you're in a coma, that even if you had a preservation team that could literally instantaneously preserve you the moment that you've been declared legally dead, it still wouldn't be good enough because the window simply doesn't exist. The window is negative. The window ended before the person's heart stopped. And that this is probably the majority of cases, because the majority of cases people die is from total organ failure in a hospice scenario. And the other ones aren't much better. It's a type of sudden death where you're completely lost. Maybe in theory there's a window, but because it's sudden, you're never going to be able to get a preservation team in quickly enough to preserve them until after you've had hours of damage accumulated. So the majority of deaths I don't think will work with the reactive preservation model, which makes sense. I mean, if you went to a doctor and the doctor told you, oh, I've got bad news for you, you need a heart surgery, and if you don't get it, you're likely to die. And you told that doctor, well, okay, well, what do I do? And the doctor's like, don't worry about it. And you're like, what do you mean? Oh yeah, just go home, don't worry about it. And you said, well, what do you mean? And the doctor's like, oh, just wear this bracelet, and then after you die, then we'll try to do the heart surgery that you need. While it's true that in some very, very rare cases, that might actually work, in the vast majority of cases, it's simply not going to ever be able to preserve that person properly, and ever be able to, if it was the case where you did a heart surgery, you could actually see afterwards whether they survived it or not, that you would be looking for a new doctor, right? And I think that brain preservation is finicky enough and complex enough to get it right, that it demands a similar level of preparedness and scheduling as any other type of major surgery, which is unfortunate. When I started the brain bank, I was hoping that it would be the case that that there is a window in general. You know, maybe 10% of cases there wouldn't be a window, right? Like really extreme comas. But I was hoping that if you die in a normal hospice setting with total organ failure, I was hoping maybe there's like 2 hours or so after that, where if you do your preservation properly and then you do electron microscopy and light microscopy on the brain samples, they look great, they look phenomenal. I was hoping that would be the case. And I did this brain bank because I thought genuinely that might be the case. And I had good reason to think that could be the case because human brains genuinely are tougher than animal brains in a lot of respects. So for example, like a rabbit hippocampal slice, you know, if you can keep them alive in artificial cerebrospinal fluid under the right protocols, but if you just look at them wrong, they die. Whereas a human hippocampal slice, you know, you can still have neurons that are alive like 96 hours after death. It's crazy. But that makes sense. Humans live a lot longer. We're omnivorous. It makes sense to build us out of the good stuff compared to mice or rabbits that are only going to live a short amount of time and then reproduce and die. Like, why waste the quality stuff for their brains? But just seeing how extreme the agonal phase is and seeing how difficult it is even to get basic stuff like perfusion right. If someone's died and there's, you know, a reasonable amount of time that you can spend to try and gain vascular aspects, and then seeing what the actual images look like, it is they would not qualify for the brain preservation prize in most cases. And that's just how it is. I mean, I'd be saying something different if I'd gotten different data from this. And I certainly wish that it would have been different, but you know, that's what I've learned. So what does that mean? Like, where do we go from here? I think that it's possible to articulate these standards as I've done before. I think the standards are clearly at a level where if you could meet those standards, you would have an overwhelmingly powerful case that you can preserve memories. And I think that it is possible to meet those standards. It's just difficult and annoying to do. And I've become convinced that the only way to meet those standards is with death with dignity protocols, or also known as medical aid in dying protocols. So this is where you have a much more tightly controlled death that's triggered fairly acutely through drugs, which is certainly a controversial medical procedure, but is also legal in 9 states. And because the MAID protocols do not have this long agonal period, because the death is very acute, it's essentially respiratory failure at the end of the day. I'm quite certain that there does exist a window in those cases, that if you die from respiratory failure caused by death with dignity drugs, it's very much more similar to the cardiac arrest case, where you do have an actual window after the person has been declared legally dead, where if you were fast enough, you could get high quality brain preservation. And so given that the window probably only exists reliably with death with dignity, I think that has to be the core of any serious preservation protocol. And then building on that core, we need animal models of this. We need testing under real world conditions. We need peer review. Basically, the minimum set of standards I think that makes sense for any sort of brain preservation protocol is you ultimately take pigs, you do exactly the protocol that a human would go through, right? Including the time delays required for declaration of legal death, including the exact drugs that would be used in a MAID protocol. You perform your preservation under the exact circumstances that would be done on an individual, and then you gather electron micrographs. You do that like 5 to 10 times at least. And if you had extensive electron micrographs from pig brains that were done under these types of real world settings, and you showed that those electron micrographs are consistent with in-ground preservation, meaning that the synapses are preserved, I think you'd have a very strong protocol that ought to work on in a human case. So I think that's doable. So it does give me some hope that it won't be long before we have a protocol that works for humans. And to anybody trying to loosen these constraints or to try and deal with protocols that we already have, I would simply recommend, do these experiments, right? Like there's nothing stopping Alcor, for example, from running their own human brain bank. I mean, people donate their brains all day long. And if you preserve a brain under those conditions, you can still do it under exactly the conditions that a normal cryonics protocol will be done. but then there's no ethical obligation whatsoever to not destroy that brain, right? So, why not for every like 10 people that are preserved, right? You do one brain preservation in a brain banking context, and then use every destructive imaging technology we have to investigate what that level of preservation is. And then also mirror that on animal protocols. Mice are very cheap, rats are very cheap. You can take a rat, you can cause death through a simulated MAID protocol, and then you can test, what are the different windows that exist for getting a good preservation. You can take a rat and you can do the type of preservation protocol that's done with chronic patients. Try it, like have a rat, have it go through a simulated agonal phase, let it sit there for an hour or two, as is common in many chronic cases, then try and preserve it, even if it's just with fixation, right? Just see what the state of the brain is even after that. And I think that evidence is the evidence that we need to really clarify these protocols. And if you can show that it still preserves the quality well enough that it could win the brain preservation prize, then that would validate the protocol. But if not, then it's, that at least demonstrates where it falls in terms of the hierarchy of protocols to do preservations. So it's somewhat unfortunate. I wish that this brain banking project of mine was in some ways more successful. Like I wish that I've done about 7 brains, and I wish that all 7 of them looked great. Even if it's like 2 hours and you died from like sepsis or something, you can still get a good preservation. And when I started this program, I don't know, maybe I was like 30-70 that would be the case. And now that I've done it, and again, this is like one of the first attempts to actually see what human brains look like under realistic conditions. It makes me kind of sad. Maybe it was obvious in retrospect, but nobody generally looks at what the conditions of human brains are after they've gone through an agonal period. So it wasn't clear which way this would go. But given the knowledge that I've learned, I think that these are the main takeaway points that we can get from this. And that's all I really have to say. I'm more than happy to just have questions now, I think, and or debate about consciousness.
Thank you very much, Robert. I understand that after the things that you said, the discussion could go on forever. And I see that there are many comments in the chat. Now I'm kind of concerned about the Zoom cloud storage limit, because I think we are already likely to have exceeded it, and they will tolerate a little overrun, but not that much. So I do plan to stop the recording, let's say in 10 minutes. It's dangerous to go over that, we risk losing the video. So that let's start with the most interesting questions in the chat. I think the first was David Wood at about not this, yeah. If you read the chat log at 18.48, and David says, when people donate their brains for preservation, are they generally aware that memories might subsequently be extracted from their brains about activities that are unsavory, illegal, unethical, etc.?
So the question is, let's suppose this technology actually works, and you're contemplating what revival looks like, which is of immediate concern to someone being preserved, because from their perspective, like, they're going to be dealing with the consequences of this literally the next subjective day. And they're considering, well, what if my memories were read against my permission and I have some tribunal accusing me of crimes? And so, I do get revived, but now I'm suddenly exposed to all sorts of legal problems. So I've thought about this a lot, and I would argue that we already have a pretty reasonable tradition for addressing this case, weirdly enough. If you look at the United Nations conventions on human rights, one of those is that a person has the right to be accused of crimes only like in the context of their time and of their society. So you can't just pull, like the United Nations Convention on Human Rights, very interestingly, already says that if you could somehow magically pull someone from the past, that it would be unethical to put them on trial for things that have subsequently become illegal, but that were legal at the time they did them. So at the very least, things that become illegal, I think if we hold to that United Nations Conventions on Human Rights, you don't suddenly get tried for them. Like you could imagine a situation where you wake up and it's like, oh, well, you killed animals and ate them, and now you're like guilty of a lot of crimes. I think you'd be able to rightly say, you know, look, that was legal when I was alive, so too bad. Now, if you've committed a lot of crimes that absolutely were illegal while you were alive, and you just sort of got away with it because nobody caught you, it's a much more interesting type of debate. Ultimately, the criteria, I think that a person being preserved should be able to specify the criteria under which they would be revived, and in general their minds should be private to themselves, and all that's covered by the United Nations Conventions on Human Rights. But this is one of the reasons why I think the control should be part of the person. Like you get to the future gets to decide how they want to treat you, sure, because they have the power, but you get to decide whether you'll let the future like access your brain or not, and the conditions under which that would be the case. So that's a long-winded way, but I think that's at least a serviceable way to think about this stuff. And also you'd have statute of limitations anyway. Our legal system is not totally garbage when it comes to having thought about some of these issues, because you get them anyway with long-lived people, estate planning, all kinds of stuff.
We have this question from Mike Perry. Do you think the brains you reported on at the 2015 Alcor conference preserved by AESC, preserved memories?
Yeah, because I mean, they preserved essentially all the proteins in the brain. They preserved the entire connectome. So they either preserve memories as neuroscience understands them, or neuroscience is like so catastrophically wrong about how memories work to the point of being like fraudulent. And I'd question how we even have done some of the experiments we've done in neuroscience that manipulate memories at this point. So yeah, short of like discovering a new biological, like the type of evidence that would convince me that ASC doesn't work would completely upend modern neuroscience and be extremely unsurprising, or it'd be extremely unexpected. It would have to look like there's an entire class of biological macromolecules that we've missed entirely, that we've never discovered up to now, that like non-redundantly stores long-term memories, that survives all the stuff that can happen to brains that are very disruptive to brains but don't erase memories, but that for some reason it's uniquely vulnerable to aldehyde fixation, is totally wiped away by it. The only candidate I can personally think of that would fit that would be if for some reason the brain already uses aldehyde fixation itself to modify proteins and store memories. That would be the type of thing that could actually get blanked out by aldehyde fixation, because it would render everything ambiguous, and would still be durable enough to survive all the things that happen to brains that don't erase memories. But there's no evidence that the brain uses aldehyde fixation in that way. So there's things that would convince me, but they're extraordinarily fringe, to the point where I don't think any serious neuroscientist is actually proposing a theory of memory that would be in contradiction to engram preservation. So yeah, therefore the memories that were in the pig and the rabbits are still there, although inaccessible.
We have the question from Gunther at 1907, and after this we will have time for one more question. So Gunther asks, 1907, if there is a way to drop an entire brain's temperature by delta T degrees using electromagnetic and magnetocaloric effects, is there a formula that would tell after how much time the memories are being irreversibly lost?
So the magnetocaloric effect is only, they've only ever been able to get it to work, I think, for like 1 degree Celsius for like extremely contrived systems that are like really, really far away from biology. And so I don't see any practical way to make it work for brains. And then I guess, what is this question asking? It's like if you just dropped the temperature of the brain quickly, like it would still freeze. And so it'd cause a lot of damage. But it's not so much like, what's the graph of when the memories would be destroyed? It's more like, what artifact are you creating from the process, right? Like one way to avoid the dynamics of this is like, what artifact would be created from a process like this? It would be a frozen brain, like literally frozen. And is it likely to conform with neuroscience theories of memory? And the answer would be no, because it's been like utterly crushed by ice formation. But I don't know if I'm answering the question. Does this make sense to you, Ken? Does this question make sense?
If I may talk about it, the question is more like when the oxygen is cut off, it appears that it's important what's the brain's temperature at that specific moment. If it's 36 degrees, it will survive longer than if it's a 35 degrees or 34. And if you could comment on that.
So there's this fascinating paper from only a few years ago. Let me talk about 2 papers. So first of all, one of my favorite papers, just because it's so crazy, is from the 50s, and it was the Navy. And this doctor at the Navy just built this like necklace thing that he put on these sailors that would just choke them to unconsciousness. And they just ran this thing and choked out a bunch of sailors and just kind of wrote what they saw. And that's the paper. It's wild. But anyways, what they learned is that you become unconscious after 30 seconds of not having oxygen to your brain. But the paper that I think is really useful here is there's one that came a few years ago where they have these pigs and they disconnect the brain circulation from the body circulation. So the brain and body are totally separate, and they put the pig on a respirator so the body is able to breathe. And then they cut off the circulation to the brain. So this was supposed to be a test of stroke, essentially. And so the brain's not getting any oxygen and not getting any blood flow, but the body is still on a respirator. The body and the heart's still beating, and the body is still getting blood flow. And they showed that you can actually go for 30 minutes of warm ischemia with these pigs with no blood flow to the brain and still recover them. Like they actually stitch up the connections again and restore the circulation between the brain and the body and close up the pig and have it walk around and do its thing. It doesn't seem like it has neurological deficits from this. So, that really surprised me because it pushed the limits of recovery from ischemia almost double what I personally knew of before. So I think that's a good starting point for normal thermic ischemia. And then if you want to know about like lower temperature, there's an extensive literature on deep hypothermic circulatory arrest that kind of shows that if you can cool to below 20 degrees C, then you get like 90 minutes to 2 hours at least of time where people are viable. So those are the 2 important data points that I know about it.
Okay, we're now running over storage. Robert, can I ask you to switch your screen off so we can see everyone? That's it. Oh, many people are still here. Things were very interesting to discuss. So I would like to thank all speakers and all attendees of this Terasem Colloquium. And I look forward to seeing you again at the next one, which is scheduled to take place in July.